This is a penultimate day. Ultimate day is coming tomorrow. I hope the course is also ultimate. So maybe we can have any inputs from your side about the course, because I am meeting you for the first time and I was thinking we should have had a ice-breaking session also, because I don't know some of you. So some of you seem to be into biophysics. Am I right? Maybe quick introduction karenge. I should know each one of you. Yes. I probably know him, but still. Keep sitting. You know?
Just completed my PhD.
Completed. Oh, nice. टॉपिक एक टॉपिक बताएंगे ना क्या है?
Sir, my topic was design in vivo in silico designing and the synthesis and validation of novel manufacturing to ameliorate Alzheimer's disease.
बैठे रह सकते हैं ना? You can keep sitting. Guneet Kaur. टॉपिक बता सकते हैं, मेरे को थोड़ा रिलेट करने में इजी होगा. If there is something I am going to tell, then I can relate better. Neuropathic. Which model? Liquid biopsy.
My name is Sandeep. I'm from I'm a first year PhD student and main program that we are working on is
Neuro?
neurodegeneration.
Viral. Yeah.
So in GIAN, currently I'm teaching from pharmacology from Department of Pharmacology and currently my research work on multiple forms.
Good morning, sir. Myself, Tarun Pandey. I have joined PhD in Central University of Punjab from Dr. J.B. Medicine is my supervisor. And my area where I will be working on PhD is neurodegenerative diseases.
My name is Central University of Punjab. I am from the second year and my topic is Parkinson's.
So it seems like there is a dress code from Central University Punjab. Black. Yes. Right. Okay. थोड़ा सा ऊंचा बोलना है ना. Topic.
It's in the founding stages, but I'm planning to work on neurobiology.
My name is Anitra and I'm from the Department of Physics and I'm working on Parkinson's disorders and cell management. I'm half of the Department of
Keep sitting and then introduce yourself. Na?
Good morning, sir. My name is Varsha Bharadwaj. I am from Naipur, and I am under professor.
Naipur?
Yeah.
Dr. Sharma's lab, no?
Yes. And currently working on Alzheimer's disease. Good morning, sir. I am Anvi Pandey and I am from Naipur. I am also working under Dr. Sharma and my research project is on the Alzheimer's disease.
You will have to introduce for everybody. Yes.
So my name is GIAN. I'm in my last year of masters in the Department of Biochemistry.
So she will be working on something related with brain, but she doesn't know as of now. Yes.
My name is and I am from Department of Biochemistry and doing my master's.
My name is Nisha.
ऊंचा बोलना.
My name is
Topic of your PhD, you can tell. Huh? So you can also introduce yourself. So I am using this opportunity to know you all. That's also part of this course, that we should be knowing you. Maybe we'll come back to you, yes.
I'm Chaitanya, I'm from BITS, Hyderabad.
BITS, Hyderabad, wow.
So we are working on organelle interaction, ER mitochondrial peroxisome interaction, including chronic ear stress in areas.
ER and peroxisome interaction. Good to hear about peroxisomes after a long gap. ER and peroxisome.
ER mitochondrial peroxisome.
Very nice.
Sir, myself is Vikram and I am from Dean's University, pursuing my master's second year.
Jaipur?
Yes, sir. Under the coordination of Dr.
Khatri.
Khatri, sir.
Oh, Khatri.
And my topic is related to neuroinflammation in Parkinson's.
Great. So I didn't know Dr. Khatri student is also here. Great.
Sir, my name is Abhishek. I have done my master's in stem cell UTC engineering from Punjab University, and my dissertation topic was effect of preconditioned secretome on depression mode.
So maybe the last two can also introduce, and then I will start formally. Louder.
My name is Nitya. I'm working with Professor Rajat Sambali. What I'm focusing on is educated work on low and associated manifestations in neuroscience. We are trying to understand the stress-associated manifestations and developing a therapeutic targeting ENS and combating CNS symptoms through it. That's where it's from the
So Ramya, you also go ahead. All right. So I'm one of the course coordinator with Dr. Tanzir, you know that, and I've been in Punjab University for last maybe 26 years now, and probably we'll be completing 26 years in another couple of days on the campus. So I've been trained at PGI. I've been pursuing an interest in neuroscience. I'm also a student of neuroscience only. I think this, I said, we still don't know much about neuroscience. All right, I picked up a topic which I don't know why I picked up, but I think it's useful. That's what I will say. So maybe a year back when the proposal was signed, maybe we had a different thought. Now the thoughts also keep evolving. So I'm going to talk about flow cytometry and how it can be applied in neurobiology. So I've put few applications of neurobiology, but all the applications that I'm going to discuss will actually apply to neurobiology also. So how many of you have used flow cytometry in your experiments? One, good. One hand goes up. I think, yes, two. So maybe few more can use it by the time we close this discussion. I think it is a handy tool to use for neuroscience also. There are few things that where it provides extra strength. People in, I would say, cell culture would use in and out flow cytometry, but using tissues also you can do a lot of things using techniques like flow cytometry. So just to keep everybody along, the word flow cytometry comes from flow is fluidics. Fluid is very important component of this. Maybe in theory you read it in early classes also. Cyto means you're looking at cells, and metry means measurement. So this is something that we should be understanding. This is about a bit of history. Cells we saw for the first time, Leeuwenhoek. Then first apparatus for detecting bacteria using this kind of mechanics was in 1947. So they were trying to look at bacteria using the flow kind of mechanism. But the flow cytometers evolved around 1965 for using it for cells from tissues. And what came there was you could also add sorting into capabilities into these flow cytometers also. There were different kinds of evolution, but I would say this is the time when Hansberg from Stanford University developed a cell sorter that separated cells stained with fluorescent antibodies, and this term fluorescent activated cell sorter was introduced. So there are two terms that I will mention, flow cytometry and FACS. Both mean different things. Flow cytometry is I am just looking at measuring cells or some parameter of cells when it is moving through the flow. When I'm saying fluorescent activated cell sorter, that means the cells are labeled. So I can then use it for detection, and then I can also use it for sorting also. So there are two differences in the two terms that I am talking of. So variety of measurements can be made on cells, cell organelles can also be studied, and you can actually study any object that is suspended in aqueous environment. Does not mean always it has to be cells, and you will all agree by the time we close this. So this is already we talked. So there is a fluid, there is a cell passing through the fluid stream, illuminated light path is there, and then there would be some kind of instrumentation which we will learn a little later. So it helps us to detect cell surface markers, intracellular molecules factors, secreted factors can also be looked at, DNA content can be seen, few applications I have just listed here. Quantifying of cells can be done, I can know the absolute number, different properties of the cells. You can categorize cells and quantify them. So there could be mixed population, I can categorize within the analysis and then quantify them also. Subpopulation of the cells can also be looked at. Applications in all fields, neuroscience is not listed in this because that's a very specific domain. Immunology, hematology, pathology, microbiology, genetics, functional studies can be done. Cell culture studies we already said, toxicity studies, drug discovery, chemical engineering can be using this, biotechnology can be using this, agronomy can be using it, animal sciences, plant sciences can also use it. Alright, looking at the particle size, we talked about flow characteristics using different sizes. Bacteria was the first one we said. Look at the sizes, 0.5 micrometers, but when you go to eukaryotic cells, the size is bigger. RBC 6 micrometers, monocytes would be 14 micrometers. So these are the kind of particles that can be analyzed in flow cytometry. So one difference you see is their sizes are different here. This is the fundamental slide. This is the whole gist of my lecture today. So what you have is cell. When the light source falls in this cell, we have a possibility that would be forward scatter, there would be side scatter. So forward scatter will depend on size of the particle. Bigger the size, this forward scatter would vary. And what would be the factor on which side scatter would depend? Granularity of the cell. What kind of granularity of the cell is there would be side scattering. So I can use these two parameters only to separate population of cells. I am not doing anything but using these inherent properties of the cells to analyze cells. I don't have to do anything. Different cells, if I take blood cells, they will vary in size. We can go back to that slide. They will vary in granularity. We can separate them. Alright, if I do this, forward side scatter, I make a plot, I can get some dots, events. These are events are being picked up, the dots are indicating, these are plots that indicate events. So I can see some populations in these events also. One is a concentrated one at the bottom, one is a little more diffuse, there is another population I can see in the middle of the two also. So based on these two only, I can separate some cell types. When I am saying forward scatters, when I am saying side scatters, there has to be some detectors to pick up these signals. There will be side scatters sensors, there would be forward scatter sensors, and there would be laser beam, which will be the light source. Alright, we said all this, what we have been talking. Forward scatter is related to surface area or size we were saying, detected along the axis of the incident light. Side scatter, reflected as well as refracted light, both are counted. Related to cell granularity and complexity of the cell. Detected at 90 degree of the laser beam. So both detectors are there. So this is one illustration of this. No, I'm moving on further now. What is this picture showing you? This picture, let's look at the graph only. It is picking up fluorescent signals. Forget about side and forward scatter. What you've done is you've labeled certain molecules on the top of the cell. So there are two kinds of labels that you see in this picture, or you can see two kinds of circles at the bottom, which I can say are the circles which are representing cells. One cell population is having less expression of antibody on the top or marker on the top. The second population is having more of that. So when I separate them, based on that, they will also show size variation is also there, and there is another variable that I brought in is fluorescent signal that I can pick. So based on this, I can actually analyze n number of parameters. What would I require if I have to analyze n number of parameters? So I said, using these, what have we done so far? We had forward and side scatter. Now we have added, maybe in this picture I could say one antibody, or we can say two antibodies. How many parameters I am looking at now? 2 or 3 at the most, maybe 4. How many parameters can we analyze in a flow cytometer? Anybody who will make a guess? At one time. Any guesses on number of parameters I can analyze? Guess. Wild guesses are also okay, but I would say instead of wild guesses, intelligent guesses are better. How many parameters? It will just depend on antibodies. How many different kinds of, and the laser beams. So the capability of the instrument improves if I have more number of light sources. I may be giving you one example of a fluorescent signal using, let's say, one filter. If I add 4 filters into the flow cytometer, I am increasing my parameters to maybe 16 or 20 parameters or even more. So it depends on the number of filters I have in the instrument. The cost of the instrument also goes up once we talk about number of parameters. You can have more filters means more parameters. But the only problem would be when I pick up my labels, I have to see they should not be, we'll talk about those, they should not interfere with the signals. But the capability increases moment I add fluorescent. So when I was only talking about forward and side scatter, I was limited to the parameters. So this is just a data from forward and side scatter. I have been able to pick up dead cells, bigger cells, size variation, apoptotic cells, live cells. Another example, blood cells, granulocytes, monocytes, lymphocytes could be picked up. Size, granularity variations. So there are few limitations of, so what I should not have talked actually about fluorescence in between, but my slide came in between, so I just mentioned that. So what are limitations with light scattering? Some information can be obtained. If I have two cell types which do not differ in size and granularity, I have a challenge. FSC correlates with cell size. FSC can distinguish living from dead cells. Side correlates with internal complexity, we said this. To distinguish between two cells size, we need both these parameters to be different. I said this already, that if they are not different, we have a problem then. If these two parameters are the same, then no distinction can be made. This is a limitation of forward and side scattering. This is a plot of size and granularity from the alveolar fluid, from biopsy mice, so lot of things. But I will have to sort and pick up the population that I am interested in, and we'll come down to that again. So then we said I can pick up other parameters to analyze. It can be based on cell surface markers, which I am labeled it as cellular antigens. Intracellular cytokines could be used, enzymes could be used. Structural differences, that means some adhesion molecules. Metabolic differences in the cells could also be picked up. I can use it for analyzing metabolic changes. I can use flow cytometry for even knowing something happening in the mitochondrial environment. I can use it for study of intracellular calcium levels. A lot of things within metabolism can be looked at. And it says probably I could be just giving a tentative kind of list. 160 different parameters could be analyzed using this technique in different populations. We talked about this now. Adding fluorescent signals, you will have to sometimes fix a cell, incubate with an antibody, permeabilize that also, label, and then signal has to be acquired using whatever we said. So what is most important here? Two more important things come: fluorochromes and antibodies. So we know fluorochromes, these are the common ones. FITC you might have used in other techniques also. Phycoethrin, PERCP, APC, many others. They could be small organic compounds. Then these have to be complexed with antibodies. This is what is a flow cytometry antibodies. So we all know fluorescence spectra. I don't know how to say. Has to be excitation wavelength. With this is what we are saying of the light sources, and there has to be emission spectra also. And I'll just skip this slide. We all probably know this. I will not talk about this. This is a picture I am revisiting that. We had explained this. You are picking up signals, fluorescent signals, instead of the other parameters that we talk of. I said you can analyze n number of parameters, but this n number of parameters would depend on this: emissions and excitations. We will want them to be apart so that I can detect those. There should not be any leaching, there should not be any quenching. These are the things that we need to be careful when I'm picking up, selecting my labels. So there is a lot about this. Probably I will not talk about this. Is just picking up the emission spectra of these. So I will not prefer APC and PERCP together. Maybe I will use them which have emissions far apart. So when I am talking about emissions, look at now the complexity of the machine also improves. So I will have light sources of particular wavelengths. I will also have different detectors now. So machine becomes more complex. So what is the most important thing in a flow cytometer? What do you think would be the most important thing in a flow cytometer? Heart of the machine.
So what is its specificity, like how efficiently it can produce?
I am saying heart of the machine.
Like,
What you are saying is not incorrect. I am saying heart of the machine. Where do you think is the whole emphasis on the machine?
the light source,
I was thinking something different, bit different. Fluidics is the most important actually first. Then comes the optical system. So light sources and the detectors would be the second most important. Fluidics is the most important, and that's where also the problem lies also when you run the flow cytometry. Yes.
Sir, 160 parameters in total or in one run?
So I would say the one run parameters, usually people have gone to something like 16 or 20. Could be a little more in theoretical sense, or maybe some people have tried that. But I would say more than 6, 8, 10 people don't go, because you don't have those probes that efficient which will be segregating that. But people have gone to 16, 18 parameters at one go, using different light sources. Yes.
Sir, and the parameter which we can analyze it will depend upon the laser source we are processing.
Definitely. So I can have UV, red, green, blue, whatever sources I use will add to the capability of the machine. So when I am buying a machine also, I decide then which light sources I have to use. Depends on how much I have in my wallet. I can increase the capability if I have more money.
Sir, can you go to FITC, PE and ABC?
This or earlier one? Earlier? So what I am saying here is, this is just telling you about excitation spectra of these. So excitation for each one is at a different wavelength. If I say FITC is getting excited at 488 nanometers, if I go to APC something like 600, that means Probably here it will be green filter. Here it could be red filter. I could be wrong, in telling you the filters. Maybe I'll show you a table which has the filters then. So based on this, my excitations vary and my emissions will vary based on what probe I am picking up. So most common one is FITC. PE is another common one that people use. So you will get antibodies that are already labeled with these from commercial sources. So you can have primary antibodies labeled with this. You can have secondary antibodies labeled with this. Both are available. So if I have to use same secondary body, same probe, I can use secondary bodies that are something like ELISA also. But ELISA doesn't give you primary antibodies already labeled with a fluorescent signal. So you will use secondary there. But here you can have primary antibodies also available. So that means one step. So most of the times we'll do one step. We are not doing any secondary antibody into this. A possibility of that is also there. So I said this is a typical flow cytometer. Now you can see forward is being seen, side is being seen, fluorescence 1 for FITC detector, FL2 for PE, and then there is FL3 for other two dyes also. So I can also have more number of detectors depending upon what signal I am using. So I have already talked about this. Maybe we will see this slide again. You can go to BD Biosciences website. It tells you what dyes I can use, what are the emissions, what are they, and it will even guide you which of these will not interfere with other signal also. This is another pictorial representation of the same kind of things. Blue-green laser. These are the dyes that can be used. Violet laser can be used. Then you have a capability of using Pacific blue, M cyan. Red laser, you have a capability of this. This is what is adding to the number of parameters you can analyze. I have not added one more filter into this, that is UV source can also be added into this. That's also useful, but it becomes more expensive also. So this is now dyes that earlier we were talking about molecules or capabilities wherein we had antibodies labeled for detection. Now we are saying molecules themselves can also be used. We don't need to go for immuno labeling of cells. Acridine orange, RNA DNA binds, discriminates between them, lysosome labeling, amino actinomycin D, DAPI for nucleus chain, you will need UV here. That's where it is adding a capability here. Propadium iodide, you can use with red laser and a few other. So likewise, I will not highlight on all. Hext is common one people use, but you need a UV source for that. So this is where you will have to decide the stains. So if I am using DAPI and propadium iodide, so if I have to use, I don't have a UV, I probably cannot use two of these filters. I probably will have to go with propadium iodide then, or likewise. Reticulocyte labeling can be done with another dye. So look like the capabilities increase. This list can go on further. Maybe I'll just pick up one or two here, not all. It is giving you a fluorescent list of fluorochromes and it is telling you what is the advantage of each. For example, Lexaflor, it says substitute for FITC, better stability and intensity. So based on these kind of parameters, you will decide which one is what I want to use. Rhodamine green, let's say, I will say, again the same kind of properties. Maybe we will pick up something for CP, long red dye. So if I have to go to red range, I probably would use this. So I'm just giving you an idea, but then you can, once you dive down into it, you will find better answers to what I'm sensitizing you with today. We can also have specialty dyes. The earlier list we did not have specialty things. I was actually looking at giving a thought that probably something is missing in my slide, but this is what I wanted to talk. If I have to do calcium measurements, I can do these two dyes. If I have to look at mitochondrial membrane potential, JC1 is also used, which is there in the bottom list. There are others in this list also, reactive oxygen intermediates people might be using or could have used also in the past. And cellular membrane potential things, other things. Okay. For Golgi apparatus we have something different. So tandem, you can use tandem fluoro probes for the reason some of these fluorochromes would impact the other. So you will have to find which one worked together. That will be tandem fluorophores, and these are some of the examples of tandem fluorophores. I am not getting into this as of now, but this is a complete list. This is from BD website. BDs are market leaders. I am not marketing for BD, but broader information from there. So you can see the kind of lasers, you can see the excitation lasers, you can see the fluorescent channels can be used, and the fluoroprobes that BD recommends for that. So this actually is a serious component of analyzing parameters in a flow cytometry. If you attend a week-long workshop on flow cytometry, this is what they are going to teach, mainly how to decide the fluoroprobes or how to analyze the data. Running is not that difficult part. So my idea is to sensitize you, tell you in brief. Maybe some of you can use it. It's not a difficult technique, and maybe I'll tell you some applications which I have not in my list also today. How can I present my data? I can present my data like this. What this data shows is number of cells and the events, simple graph. So it tells you me two populations, one which has low CD count, other has high CD count, and the one which has low CD count has more events. Likewise, I can now analyze two parameters. When I am analyzing two parameters, I can have CD8 versus CD4. So my plot can come out like this, and I can say at least there are two major populations which can be differentiated here, this and this. So there are different ways of presenting this data. Histogram, one we saw earlier, 2D plots, the one which showed was dots. We can have 3D plot, CellQuest is the name that is given. I can have 3D plot attractors as another way we can do that. This is one of the instruments, Fax Caliber. We have one in our department called LSR Fortessa, which has 3 lasers I guess, and probably we bought it maybe 10-15 years back. So system has this fluidics, it has optics, it has electronics. This is majorly the components. I will not spend too much time on this, but I will just go through. I said heart of the machine is fluidics. Why fluidics is important? I want to answer from you. I did not emphasize why it is important. I forgot to mention one thing very, very important. So what the role of fluidics, he said that fluidics role is to have a system that there is one cell passing through, or you are able to analyze one cell at a given time. That's the whole idea. If the cells adhere to each other, probably the purpose will be lost. So there has to be certain parameters, and that is why we are saying air filter, that means airflow is also there. Sheath fluid is the fluid in which you are carrying the cells, and they will pass through a narrow capillary, which is where you are analyzing the sample. And whatever is then passing through is then collected through the waste. But if I want to improvise the capability, I can not let the waste go. I probably can use them as separation facility. This is what is fluidics actually now. So sample is shown in a reverse order. You can think that the picture is in the other order. You have a sample, but when it passes through this capillary, they are all in as single cell. So this is the most important part. That's why I said heart of the machine is the fluidics. And you spend money, recurring cost is also on the sheath fluid that in which the cells move in as a stream also. So excitation lasers and rest of the things are all part of it. Anyway, all right. So I can vary the pressure. You can have a machine with low differential pressure, you can have high differential pressure. The rate at which the cells move will be more in the case of high differential pressure, but in Each of these cases there has to be single suspension. In this case you only can see one cell, you can see two cells, but advantage of that would be I probably would be able to capture more events. So optics we all know what will be there, detectors and all. We don't want to get into the instrumentation business because that's something I don't want to spend time. And this is how the optics looks like, and that would be so. And there is a lot of emphasis on optics because look at the kind of detectors that you are using. Look at the kind of sources of light you are using. So the optics also has to be very, very important, and that's why the machines are expensive. This we already talked. Maybe this is the architecture of one of the instruments that we can talk. So you can see all these filters that are picking up the signals. FL1, side scatter, FL2, 4, I showed you in the early picture 3, and likewise you have excitation and side forward and side scatters. Measurements, we talked about these. This is just an example of picking up cells what is the cell being picked up? CD14 negative, CD18 plus. That means antibodies would be binding, these red ones are the antibodies that are binding to CD80 plus. CD14 is green. So based on these I can separate CD14 versus CD80 cell types. So already we will discuss this figure. I can have 4 different populations based on these fluorescent signals. This is the dot plot we were talking. So this is CD14 versus CD80. So I have seen cells which are negative for both, they will all come in this population. CD40 positives are coming here, CD14 positives are coming here. The ones that pick up both could be in a broader population, that is somewhere here. Histograms could be there. For these same data, this is just CD14 in the left and signal being picked up here. I think there is a problem in my picture. I should have had the other picture, same picture got pasted here anyway. So one more thing in flow cytometry is gating. This is one picture, forget about what my circles say for a minute. What I can do is now, this is my population coming from forward and side scatter. I can further analyze these populations, this population and this population by gating these, which is a software based application. So I am gating these and further analyzing these populations because this might have subpopulations within itself. So that is the advantage of having both gating. So gating also becomes a very, very important component in flow cytometry. So you have seen R2 has been separated into populations. Likewise, R1 has been separated into different populations. You can do gating and separate them into different populations and then analyze them making different kinds of plots. So what all applications can come now? Phenotype analysis, intracellular analysis, DNA analysis, cell function analysis. One thing I have not talked is cytokine bead arrays. How many of you have heard of this? No. So I said you don't have to analyze cells, you can analyze any kind of particles. And that will be what is called cytokine bead arrays. That, as the name suggests, I can analyze cytokines. How do you normally analyze cytokines? How do you normally analyze cytokines? कैसे देखते हैं cytokines हम? Yes. ELISA, basically you do ELISA. So if I do multiple cytokines in a given sample, this technique will be useful. Little amount of sample, I can analyze 10 parameters, 20 parameters. That's the advantage. We'll come back to this and explain that. Cost is a little higher because you are using more antibodies also in the same sample. So you can't expect cost would be down, but sample saving is the most important here. Intrinsic, we talked about this. Extrinsic, when we are adding agents to analyze it, it will be called as extrinsic parameters that can be analyzed. So we talked about all these. This is kind of still a repetition. This also we have already seen. It's in different kinds of population. Phenotypic analysis, we talked about these. I can analyze lymphocyte phenotyping can be done. This is very useful in immunology. Yes. Which one?
Can you comment on how the cell's intrinsic fluorescence would impact the extrinsic fluorescence? Suppose if the cell has its own intrinsic fluorescence.
You're saying the intrinsic fluorescence, some of the cells might have their intrinsic fluorescence, but that signal will be very weak. That will be very, very weak. You don't expect that to be there because you are using labels which are much stronger and exciting at specific wavelength. So I don't expect that to be part of it. Yes, there could be quenching, but those kind of issues can be sorted out. Maybe we can have a detailed discussion on that. If I have my slides, I will show you that, but otherwise quenching is also important because some signals can even mask your signals also from the cell itself. Or each label can also mask each other. What you are saying is the inherent fluorescence, but eukaryotic cells you don't have it. You will have that problem mainly in plant cells because you have more fluorescent pigments into plant cells. So you don't have it in eukaryotic cells, that bigger challenge. Minor will all be taken as baseline. So not an issue to worry about in animal cells. So I can do different populations, all these populations. I don't have to tell you that, you have understood that. I can then look at I have been able to analyze so many different parameters from, so immunology labs in a hospital will be using this. All these big hospitals, immunology labs will have flow cytometers to analyze these kind of parameters, the populations of cells, because the treatment depends on some of these. Fine, allergic reactions, you want to analyze the cell population, just not antibodies. Intracellular analysis, I talked about these. When it comes to intracellular analysis, I will have to do two things: fix the cells, permeabilize the cells. Same kind of techniques that I do with histology are used here also. Histology or immunohistochemistry. We use a detergent, could be tritone, could be anything else. We will also use formaldehyde, 4%, for fixing the cells here also. So in the tube you are fixing the cells, you are permeabilizing within the tube. This is a cartoon to illustrate that. There are membranes, now the antibodies are entering and you can pick up the signals from within the cell. So easier is always cell surface. You can do it within the cell also. So a lot of things, cytokines I said could be detected intracellular also. We might have an interest to know how much is the production of cytokine. So there you will need permeabilization agents. This is an example to tell you that. So I can stimulate the cells to produce some cytokine, analyze that, and it says formaldehyde, glutaraldehyde could be used, permeabilization, and then you can do staining and then pick up different kinds of signals. All right, I will skip some slides in the interest of time. I can also analyze signal transduction parameters, many of them. All right, intracellular staining in activated lyse blood cells. This is talking about intracellular stains. That means I am picking up what is the signal being picked up. If anybody can read the slide, my slide. It says intracellular signal. STAT3 is a transcription factor. You are using a phosphotyrosine label STAT3 antibody to pick up how much is STAT3 expression inside the cell. So I'm just giving an illustration. You don't expect me to go deep down into it because that will take a lot of time for us to pick up what this each figure interprets, but I just want to give you an idea that I can pick up intracellular molecules by permeabilization and fixation, and in this particular slide it has picked up STAT3 signal using a phosphotyrosine label for that particular antibody. This is most important. DNA analysis can be done. Neurobiology will have application. Cancer would have more applications because cycle is more important in there. So same ethanol detergent, you could have nucleic acid dyes. These could be the kind of dyes we could use. You all familiar with this dye. When I do the analysis, I can pick up cells in different stages. G1 has the maximum DNA content, followed by S, G2 or M phase. So this could be the picture that I can put it in other words, and I can analyze different cell populations. Within this entire population, I can pick up sub G1 stage cells from that population by using this gating. So I have a population, within population I am screening a subset of populations. Alright, we talked about some of these, and I'll come back to apoptosis, which is again where it is commonly used. Cell proliferation, BRDU stain is used, propadium iodase is used, calcium we already talked. Intercellular pH determination can be done by SNRF1, oxidative metabolism, TCF, DA, membrane potential, we talked some examples. How many of us are familiar with annexin 5 stain? May not be. In apoptosis, membrane asymmetry changes. So what you see is, during apoptosis, what you've seen is the red dots have moved to outside. So now they can bind to annexin 5 FITC conjugate, and that can be a new signal that I have emerged now. Annexin FITC stain can pick up these apoptotic cells. Adds to my strength in analyzing these parameters. Look at what we have got now: live cells, necrotic cells, and apoptotic cells. We have been able to analyze cell death using some stains. This is FITC stain, annexin 5 stain. Using PE, some populations I'll not get, but I just wanted to give you an idea. You can also look at mitochondrial membrane potential using JC1 stain. So you will pick up signals on what is the membrane potential. If it's changing, JC1 stain would vary, and that also would be picked up. Proliferation assay, I said BRDU stain can be picked up. This is where I wanted to spend time with you for a few minutes. Now, another technique which is powerful and handy is cytometric bead arrays. I said cytokine bead arrays, but it should have been cytometric bead arrays, because mainly we are using for cytokines. So you have a bead which has antibodies. So antibodies are now capturing their antigen. So this is the antigen, which could be cytokine, picked up by this bead. What happens in, so how I am analyzing multiple parameters? This is the answer. You are using beads of different sizes. Size differences would be picked up. Different fluorescence intensities, different colors of beads could be there. You are adding capabilities. I could analyze 8 different parameters for cytokines using this. Multiple analysis simultaneously, reduced sample volume requirements, reduced hands-on time, wide dynamic range of fluorescence detection, no problems in terms of interference. This is a data from cytokine bead arrays. So many cytokines analyzed. This is another way to plot that same data. Anyway, you can also make standard curves using this, not getting into that. You can have, this is another data from cytometric bead arrays. Open configuration. I can decide what should go on my beads. Not only the pre-made ones. I can have 30 different parameters, and I can use clustering based on red and NIR fluorescence intensity and dual lasers and things like that. Anyway, we've talked about this. I don't have to say much. We can improvise this technique a little more. All right. This is what would be another capability in flow cytometry. I don't throw the stuff that comes out, separate that into different populations based on voltage. The ones which have no voltage are in blue, or no response to the voltage are going straight. Others will be deflected based on the charges. You can do the sorting. People use it, the cost adds to the machine. So if I am buying it for 1 crore, it will maybe go 1.5 or 1.78 crore. So you will spend more money. But why would you need a cyto sorter? Because if you want to use the cells again for doing something, then you need a sorter. Or your purpose is to sort the cell, then you will be using it. Otherwise, if my purpose is only analysis, why should I spend money on sorter? All right. Can I make the technique even more simple? Yes. Magnetic sorting. I can have magnetic beads labeled with antibodies and do the sorting. There are a lot of techniques available where you can use magnetic sorting. Magnetic sorting could be a step before flow cytometry also. More cells in a population, I want to enrich the population, I will use magnetic sorting. In this particular case, it's saying sperm sorting is being done, but I can use it for any kind of cell. I only need antibodies that or something that my population can bind to a magnetic field. So what it says is magnet, it holds the population, I demagnetize the environment, that same population is left out. So I can separate cells using this magnetic, and these are less expensive also. This is where I will have another dimension added to my flow cytometry experiments. Now coming to the real topic that I had to touch upon, but all that I told you can also be used for otherwise also in neurobiology, but I will repeat so that we have an emphasis. There is something that I will add extra. I said you can analyze any kind of parameter. You can analyze nanoparticles also using flow cytometry if I can pick up the differences there. If I have two populations, I can use, I am making nanoparticles, I am loading with something, I know there are inherent differences in the population, I can use flow cytometry, any particle. Exosomes can also be analyzed using flow cytometry. Just giving you another example, we've used that sometime back, and this is the capability. Cell population analysis, we can do that. Phenotype and marker analysis, we talked about that. Intracellular state, we talked about that. Cell sorting also we talked about that. In brain that becomes important. Why? Because we have different populations, neurons, microglia, astrocytes, few other populations. There magnetic sorting will also be helpful. I will also come down to one more technique. Receptor quantification is cell surface marker. I want to analyze the number of markers on the receptor on the cells, specific cell types within the hippocampus, stratum, and see their expression. I can do Western blot also, I can use this also. Advantage with this would be, if I am separating cell population, I will know which cell type is contributing to this. Neuro-oncology, yes, I can pick up glioblastoma cells and then analyze them in terms of cell cycle, in terms of other markers which will be specific to cancer and things like that. So two more examples I will talk to you is, one is this. So I said you can dissociate tissues, remove myelin in cell debris, separate one set of population, say CD11 bead incubation, magnetic separation. Which kind of cells will be separated within CD11B? Which cells are we separating with this? CD11B is marker for easy है, मुश्किल नहीं होना चाहिए. Microglia. So microglial marker. So you should be able to collect CD11B positive cells. Negative also I can pick because I know where the negative has gone. That will be in the first part of the sample. I can use even simple technique if I don't have a magnetic separation technique available. Look at what my right side says. Dentate gyrus has been isolated. I've just picked up from the net. So why dentate gyrus should not be asked because it can be any section of the brain. Tissue can be dissociated. Homogenization. Then you can separate them through cell strainers. They are available at different porosities. I can separate cells and then analyze those cells. And it says you can label those cells then with antibody and then rest of the things. This is, I find, is more interesting even. How many of you have known synaptosomes? If I homogenize a cell, my neurons pinch off and form vesicles like this. So these become functional units coming from neuron. I cannot analyze neurons, I can analyze synaptosomes as a functional point of neurons. This is just an example where they have gated the synaptoneurosome population. And the same population has been analyzed using electron microscopy also. But what I said to you, the synaptosomes, these are the circular particles that you can see are synaptosomes. So I can analyze these also using, and I can do any kind of study using synaptosomes because these are also vesicles now. They are kind of smaller cells which I can analyze in terms of calcium influx. I can do oxidative stress measurements. I can do any kind of parameters. I can look at the presynaptic terminal markers of on this also. At the same time, I can look at these populations using microscopy also. So this is a summary of what I said: cell phenotyping, cell cycle analysis, cell functional microbial sorting. We didn't touch on this. We can add few more things to improvise on the data, and it can be high throughput, purity, single cell sorting, multiplexing. All those things would be beneficial. This is all the summary. All right, I'll just say it is a valuable technique for studying cell diversity, cell signaling, changes in neuronal cells during disease, can even sort and isolate specific cells for further study. So I just wanted to give you a window to this. Maybe rest of the things you will read a little more and then maybe try to have this in your research if possible. Thank you. Any questions? Most welcome.
we have then a short break and a tea, but the tea will come after 10 minutes.
Yeah.
Okay. So break.
You can join sir till that.
So I will at the beginning speak a little bit about the imaging equipment, then about the new generation imaging probe, and then at the end show how these different kind of probes could be used to image different mitochondrial functionalities. Can you hear me well enough? Good. So, imaging equipment, you can separate maybe the non-live cell imaging equipment and live cell fluorescence microscopy or imaging equipment. So you know the electron microscope that are giving you very good nanometer, sub-nanometer resolution. Then you have also cryo-electron microscopy and cryo-electron tomography, which could give you, in case of cryo-electron tomography, also the few nanometer resolution and 3D images. So they are very expensive. Electron microscope, maybe not few $100,000 USD, but this cryo-electron tomography, which is nowadays very important, it is already 3 to 5 million USD. But this, you will get resolution, but only from the dead, processed samples or cells. And then you have a live cell fluorescence microscopy that is more relevant for the basic researchers or life science researchers maybe. So there is simple wide field epifluorescent microscopy which gives you 250 nanometer lateral resolution. The systems are relatively cheap, 100,000 maybe maximum USD. Then you have a confocal microscopes which will have improved resolution, 200 nanometers or slightly less. And they will give you also the 3D resolution, which is very important, that you don't have if you have simple fluorescent microscope. Prices are higher, of course. And then you have super resolution microscopes. So it's iris scan, though is maybe part super resolution. You will have 120 nanometer lateral resolution. SIM super resolution microscope 100 nanometer, instead it's 20 nanometers. Again, price is going up, so to get a very good state you need almost 1 million USD. So maybe this super resolution is not for everyday's work, but the confocal and the iris scan, like little bit enhanced confocals, they are nowadays quite standard imaging platforms. So this is such kind of classic microscopy platform, but nowadays you can have also the high content automated imaging platforms. They are usually not for single samples, they are for the plates. You will put in 96 well plate, it will do automatic imaging, and it has also sometimes automatic analysis system built up, so you will get numbers out. If they are good, you will get a lot of data quickly. What is the limitation? You can't play with different probes so well. Usually the lenses there are not so powerful as in confocal or as in microscopes. So, but both are good for something. So we have both systems in our core facility. This iris can do half super resolution, and also this Revit, this automated high content screening platform. So if you want to know more about these imaging platforms, then there are a lot of good sites you can find it. So the main producers of the confocal microscope, Nikon, Evident, previously Olympus and SAIS, they have all very good educational materials on these specific sites. So you can learn from there all these things you had in previous lecture, emissions, excitations, what kind of microscopes are available, what is the logic. Then there is analysis software, which is as relevant as imaging itself, because it's nice to have good images, but you need somehow to analyze these. So again, to quickly summarize, this core analysis, you can do most of simple things with it. The best known is this ImageG, FIGI. Then nowadays also Cell Profiler becomes more and more popular. They are more or less free platforms, so you can download them and use them for free. Then you have also commercial platforms for analysis. Imaris, Arivis, Visiofarm, Harmony. They are quite expensive, but sometimes they have some capabilities that are better. And also if you take Harmony, then Revity's equipment works with Harmony, and usually if you work with SAIS, then Arivis is very well analyzing the SAIS images and so on. So that commercial platforms sometimes are not bad at all. They are easier to use maybe, but they're too expensive. And then during last years, a lot of machine learning and AI-based tools or softwares, it's more that is to say tools, have become. So Elastic is maybe one of the names which is known. And also the FIGI and Cell Profiler are introducing the AI-based analysis modules nowadays. And then Meta or Facebook, previous Facebook, is supporting also development of Napari, which is for specific visualization, image management tasks. So I have in my computer only ImageG, so it's not very user-friendly, but if you learn it, then it's quite okay. So I have here time-lapse video, 2 channels. Green channel is showing the axon from the mutant Huntington expressing neuron, and the probe we are using is the calcium sensor. And we made a time course, and you see that some spontaneous calcium spikes happen. Then we have another channel here for normalization, where nothing very much happens. So if you have such image, then you can analyze, you can do a lot of things here. So one thing, for example, what I can show you, there is segmentation, robust, which means that sometimes you need to know where is the signal, where is not the signal. You will get it. You can, if you need, you can count something here, and so on. So you have a lot of very different tools. And nowadays, earlier it was very difficult to find out where is the tool you need. Nowadays you can ask from ChatGPT what kind of tool I need for that, what is the name of that ImageG specific tool, and under which menu it is. So it makes the life much easier. So why this analysis is relevant, why it really matters, this quantification? because one thing is that this time where you just showed an image, this is over. It's not anymore really relevant, because there is a lot of bias. If you are looking under microscope something, then you will always find something. Question is that relevant? Is that really there? Is it different from something you have in other dish? So you should have, from description that you had a nice cell, you should have from that numbers, how many nice cells you have, what this nice means. So you need to quantify. It's also for the for statistics, because only statistics allows you to make conclusions. Nowadays you have to repeat every experiment. How you will repeat the image, it's really not so easy. Numbers, if you have numbers out, you can repeat experiments. And you can't publish any more images without numbers in good journals, high impact journals. They always want good, statistically clear, if possible blind analysis. And if you do the drug research, biomarker research, then it's even more relevant. Then you always need analysis. Just one slide about us is that we're not just doing research in pharmacology department, but we have in our department also the core facility of biomedical imaging. So we are, we are the only core facility. We serve the medical faculty. We have the confocals, size confocals, several. We have screening equipment, and I think we are best equipped imaging department or facility in Estonia. And we are also funded by the John Zuckerberg initiative, because they considered us to be quite good, and we are the only one in Estonia really doing many different things. And we are also part of the Estonian bioimaging leading core facility in Estonian bioimaging, which is funded by state. So this is this general part. Now we are going to genetically encoded imaging probes. I'm not speaking about the dyes. In previous lecture you had a lot about dyes, but dyes are giving you some information, but with genetically encoded probes you can get much more out from the imaging. So here you see in this image the presynaptic axonal terminal. It's the dye, the red dye shows the actin, and you have one single mitochondrion, green dye, in this presynaptic terminal. So we use biological or genetically encoded fluorophores. So you know about the GFP, yellow fluorescent protein, red fluorescent protein. This was 20 years ago, you had just few proteins. Now you have a lot of different proteins. I have in this image, maybe it's just example to show that you can pick up something from blue till something to far red or almost far red, but in real life you have many more of these options. Then how to understand, because you have a huge amount of different probes, cherry, strawberry, plum, how to find out what they are doing? There is a very good database, FP-base, fluorescent protein base, where you have all these proteins, different fluorescent proteins. You have the parameters, excitation, emission, brightness, which are very important parameters. And you can also see how they have developed. So people are still using this old GFP, but others have developed these GFP further. It's the, how to say, the development tree. So they have made many probes from this GFP, mutating the GFP, and these new probes are better. If your GFP has 45, how to say, units of brightness, then the new probes, GPET, have almost doubled this brightness. And brightness is also always relevant, because yes, you have strong marker, it doesn't matter, but in many cases the problem is that your marker is too weak. Therefore stronger fluorescent proteins will give you better signal. Okay, you have a lot of different fluorescent proteins and the probes based on these fluorescent proteins. How to get them if you want to use something. Of course, you can get the plasmid expressing this, or viral vector expressing it from your friend or from the neighboring lab, but they don't have everything. So there are sources. So the one of the best source is the repository GIAN. In GIAN they have huge amount of different probes, and you can get them. They are, I think current price is $100 per one protein. You will get the plasmid. You are growing it up yourself. You have it forever. Then another thing, they are also producing some viruses, and this GIAN is not only the only site. So also there are other repositories. For example, this University of Zurich viral vector facility has a lot of viral vectors in their repository, and they are all, not all, but they are mainly, these repositories are non-profit, so you pay just to cover the costs, but they don't make profit, therefore it's cheaper. If you get the buy or not buy, you will get the probe from GIAN, it's $100. The same probe from company could be $500 or $900. And then another option is to do it yourself. The gene synthesis is nowadays so cheap. If you take the GEN script and you have relatively small fluorescent protein and reporter, then you will get it again with $100. If you want to do subcloning, maybe 100 and something, 140 is maybe currently the price. So it's even easier nowadays because gene synthesis is cheaper. Maybe it's also possible to do it cheaply here. We have to buy from bigger companies who don't have it locally cheap. That it is cheaper to do everything from zero. But then you have risks, because you have to plan it. You have to provide them sequence, and to provide them sequence, you should know exactly what sequence is working. So you should understand how this, how to fuse your protein of interest with specific fluorescent protein. You can do mistakes. We are experienced, but we are still doing mistakes. If you take from GIAN and it's published before, then it's less risky to get something which is not working. But in GIAN you don't have everything, so you can do it. You should do it there for yourself. So just few examples. That, for example, you want to know whether the protein of interest, wolframin 1, is expressed in presynaptic axonal terminals. You need what you need. You need your protein with fluorescent tag, and you need the marker of axonal terminal. How you find it? You have your protein. This you will do yourself. You will get it from someone. How to find the marker? So it's just example that you can browse the proteins markers from GIAN homepage, subcellular localization. Then you go to the cytoskeletal elements and you will pick up the marker for cytoskeleton elements, and they will mark the presynaptic axonal terminal very well. You can see it very easily. So we use this LifeAct Scarlet. You see the structure, and here it's the fluorescent wolframin. Co-localization shows that yes, this protein is expressed in presynaptic axonal terminal. So this is such kind of easy stuff, but if you are studying very small structures, then this traditional resolution is not enough. Then you should do the same using super or half super resolution system, or you can use the more complicated systems, FRET and split GFP and so on. So again, one picture I should at P4 also, that your task here is to understand whether your mitochondria, which are red, are inside of the autophagosomes. So if you have a low or traditional resolution, you really can't see it. But if you go to the iris scan to super resolution, then you will get nice images. You clearly see how this autophagosome is surrounding the mitochondria. Then one more point is that if you use the dyes, then all cells are giving you more or less equal signal. But if you use fluorescent sensors, plasmids expressing fluorescent sensors, then you will have to transfect them to your cells or electroporate or whatever. And the expression levels will be different in different cells. How you compare it if they will be different? It's a problem. So therefore, you should have something for normalization. Another plasmid expressing something, having something, another fluorophore, because usually the expression, if you have two plasmids, then the expression levels are more or less similarly going up and down, not so perfectly, but little bit at least. But the best is to have a ratiometric sensor, which means that this one sensor gives you two signals changing differently. So we have here example. We have again the axonal terminal. You see below axonal terminal, and then we have the calcium sensors to measure cytosolic calcium. This calcium sensor has two detection channels, or actually one detection channel but two emissions, but okay, this is maybe not relevant now. If you treat it with glutamate, then one signal, you see the green signal is going up, and another blue signal is going slightly down. If you take the ratio is going very nicely up. If you now compare then different cells, then these green signals are very variable. One starts from 100, another from 500, for example. Really huge variation, but the ratios are perfectly similar in these cells. So therefore, ratiometric, if you compare, if you don't just show what happens in cells, that aha, something happens, if you want to compare different kind of cells from different tissues, then the ratiometric sensors are necessary, or at least you should have another fluorophore for normalization. Okay, so now we have 20 slides about the mitochondrial imaging. So the aim is just to show you that a lot of things could be imaged. So I think in mitochondrial research 20 years ago people used biochemistry, they used respiration measurements mostly. So you see this circle here, imaging was really marginal method. If you take the papers, mitochondrial research papers now, then it's my subjective feeling is that all methods are based on imaging. So of course it's different in other areas, but it's more or less similar everywhere, that the imaging is more relevant. And currently I think imaging has been considered as a main method or group of methods in biomedical research. Everything else is, how to say, not giving so big share. So if you think what could be measured then in mitochondria, so mitochondrial dynamics, mitochondrial calcium, mitochondrial ROS production, mitochondrial ATP production, NADH, NAD in cytosol, which is affected by mitochondria, mitochondrial age, mitochondria associated membranes means that mitochondria are contact sites. And actually a lot of other things could be measured. This is just what we are measuring. You saw this video yesterday. So you have red mitochondria, it fuses with green one. So how we did it? So we have a photo activatable or photoconvertible markers. So we used here Kikume green marker, which is usually green, as the name said. So you see that blue channel is just to have a control signal, and then this mitochondria were green. Then with laser we activated one mitochondrion here, and after that, so the axon is going like that. After that it was not anymore green, but it was red. And then we followed what happens. So green and red fuse, we will have a yellow. You can see it and you can quantify it. So to have such kind of image or such kind of video is very nice, but it has no any value. The value is coming only from the analysis. If you can build up the trustful analysis pathway or scheme, then you will get very clear numbers. In this case you see that if we overexpress mutant huntingtin, then this fusion rate is going down almost twice. If we overexpress tau protein in neurons, then the mitochondrial fusion rate is going down twice. Another thing is, we were speaking about Parkin translocation. So at the beginning, Parkin is everywhere. If mitochondria will be dying, then it's in clumps of mitochondria. So you can measure that automatically as quantifying heterogeneity. So signal here is similar everywhere, heterogeneity low. If it goes to clumps, it will be significantly higher. You can make automatic analysis from that. Yes, please.
Can you quantify how much the overlap is?
Quantify,
how much the overlap, like green and red, is the overlap you see, or it's not? visual.
Yes, so you mean colocalization. So colocalization you can measure, for example, like that you will have one pixel, and is that pixel only green, is that pixel only red, or is it both? So this is such kind of basic colocalization analysis. But it is not working in this case very much, because if you will have a high resolution image, then one is surrounding another. So if you do pixel-based analysis, then you don't have very good colocalization if one is inside of another. If they are matching perfectly, then you will get it from this pixel-based colocalization analysis.
Probably like I get completely the overlap, but I want to measure how much the overlap is happening. Like, is there any software?
Yes, in all software there should be an option to quantify correlation coefficients, colocalization coefficients. What is the correlation between green and red? If this correlation is high, then your coefficient of correlation or the colocalization will be high. If it's low, then it's low, and you can compare different conditions if you need. It's a much more complicated, but in the basic is like that. So it's the same for autophagosome engulfment. Okay, then this is this one way is to quantify the autophagies, that just you have colocalization, or at least one is surrounded by another, but then you have also very specific probes, the pH sensitive probes, which change the spectra if the pH is changing. So it works very very nicely that, for example, here the mitochondria are greenish, but if mitochondria go to lysosomes, then pH of lysosome changes the color of mitochondria stain. And you will see that the green mitochondria become red. And since they are inside of lysosomes, they are not anymore the long, but they are just a round shape. It's again not exactly green and red, they are artificial colors, but it's more or less like that. And you can quantify it. If you take the pink overexpression, you have more mitophagy. If you have knockdown, if you have a kinesthetic pink overexpression, then you don't have. What for we use? We are using it to quantify the autophagy, the mitophagy in different disease models. So here you will see that it's a control case, it's disease case. We have much more red dots, much more mitochondria inside of lysosomes. And it's not just mitochondria we can visualize that way. We are visualizing also ER that way, that ER is inside of the lysosome. ER marker will be different. Theoretically, we have not done that. There is also peroxophagy. Peroxisomes are going to autophagosomes. Theoretically, they should be also, peroxophagy could be measured that way. We have not just done that. Then you can measure mitochondrial calcium. So you see here the axon again, and then you will have a mitochondria inside of axons, two different channels. If you treat with mitochondria, with something which increases mitochondrial calcium uptake, you see the signal is going up. You have much more calcium in mitochondria. And you can quantify what happens in every individual mitochondria, and then summarize it per axon, per dish or whatever. And again, you will see that in case of disease, we have much less calcium in mitochondria. So it's again not so easy. What is not so easy is to pick up a proper sensor about that. Different cells have different amount of calcium in mitochondria, and you need the sensor which is having dynamic range matching with your sample. So just a good example is that we used here two different sensors, same model, but you see here the effect is small, with another sensor effect is much bigger. So that it's not just easy, I will take mitochondrial calcium sensor, because I think you have tens of these different sensors even in AppGene. Only few may work in your model, and to find out which one works is difficult. From literature mostly, speaking with people doing that job, you can't just pick it, this is good, and without checking, is it really good. Then ROS production. You can use the Hyper 7 to measure ROS in cytosol or in mitochondria. So what we did here, it's a more complicated experiment. We expressed in mitochondria the killer red protein. Killer red is, if it's just a normal situation, then it gives red signal from mitochondria. But if you use laser to activate it, then this killer red starts to produce free radicals. And it is bleached also away. And then we used another marker, this mitochondrial hyper 7, to measure these radicals. So you see before activation and after activation, this yellow box, we activated this killer red in this yellow box. You see it became green, means that a lot of free radicals are coming from here. Then more complicated things. You can measure ATP ratiometrically from your cell. There are different sensors. 18 was one of the first. Perceval is, at least for us, the best at the moment. And there are also others maybe less used. So we express it in the axon, in the neuron. It's everywhere in the axon, also in axonoterminal. And we will get the ratiometric value, how much we have ATP here. So we treated with an inhibitor, mitochondrial inhibitor. What we saw, we have loss of ATP. So we can measure basal ATP level in the axon. And we can quantify it. Again, we have a glutamate, excitotoxicity, lot of calcium. Or we have one rare disease, SPATA5 deficiency. We have less ATP in axon. So this is a tricky measurement. What we had before is less tricky. This is already more tricky. But a lot of people use it nowadays. And how this sensor works? It is just circularly permuted variant of yellow fluorescent protein. This circularly permuted variant CP is usually the abbreviation. It is just fused to the protein, bacterial protein, which binds ATP or ADP. If you have here ATP, then you have one kind of conformation. If you have ADP, then another kind. And this is changing the spectra of the sensor. So it's the main logic, this CP fluorescent proteins are fused to some kind of sensor proteins, and if sensor binds something, you will have different kind of fluorescence. So you can measure NADH-NAD ratio using the Paradox or sonar sensors. So they are slightly different, the fluorophore slightly different, and the sensor protein, sensor part is slightly different. But anyway, they work more or less same way, that you can get numbers out, ratiometric signal, and numbers out from that ratiometric signal, and you can see, do you have a less NADH or more NADH? And in our case, we had again this disease, we had more NADH clearly. Again, this is difficult to develop method. It requires this high-end confocal. If you have just fluorescent microscope, then you can't do it. And few more things. For example, you can measure the age of your protein. Or if the protein is in mitochondria, this means you can measure the age of your mitochondria. This is more tricky, not working actually so well, but theoretically it works. And how it works? That you have fluorescent protein timers. It is green at the beginning, and if the molecule, if the protein becomes old in the cell, it turns to be red. So you have a green and red proteins, mix of green and red proteins in your cell. If these proteins are older, or this protein population is older, then everything is more red. You will have a green to red ratio for everything. So you see the mitochondria here, they are red or yellow. This means that yellow has more green inside and is younger than just clean red. So mitochondria have slightly different age. In these simple cells, they are fusing so quickly at the age is same. But if you take the long axons, then at the end of axons you may find some old mitochondria that are very red. So we had one disease model, and at the end of axon we had significantly older mitochondria than in control cells. Then the last model is that we were speaking earlier a lot about the mitochondrial contact sites. How you measure the mitochondrial contact sites? What early people did, they used this simple colocalization assay. One marker was in endoplasmic reticulum, another was in mitochondria. They said if they are together, there is colocalization, then you have this mum. But it's not working like that, because the resolution of the fluorescent microscope or confocal is how much it was, 200 nanometers. But these contacts are maybe 20, 50 nanometers. And in cell, it's heavily packed in axons. A lot of ER, a lot of mitochondria, they are always so close. So your resolution is not enough to say whether the overlap between signal is mum or just randomly close. So that this method is not acceptable anymore, and now they have developed tools how to measure this. You can of course always do electron microscopy, which gives you it's gold standard for that, but they are not live cells. You can't measure dynamics. You can't really do it quickly. So they developed such kind of probes, luminescent, fluorescent-based probes, that one part of the probe was in reticulum and another part was in mitochondria. And if these two parts were close enough, they gave you fluorescence. Or what we are using, we get easier numbers out, we are use luminescence. Half of the probe is in ER, another half is in mito. If they are becoming together, we will get fully functional nanolook, which will give us luminescent signal. And we quantify this luminescent signal. You see, disease means we have less mums in this disease. So, what to conclude from that? So that these new sensors, fluorescent protein-based sensors, are coming and coming. You can measure more and more things with these in live cells. That was earlier not possible, and that is much more valuable than to measure something in cell homogenate. And in case of mitochondrial research, it's really big development. But the problem is that this requires expensive microscopes and a lot of time. Also, to build up some new method is taking a long time, because the tools are available, but not all tools are working with all kinds of samples or cells. Why you should do it also is that you really can't publish very well anymore if you don't have imaging. In case of neuroscience, if you don't have imaging, of course you can, but if the work is such kind of work that imaging could be used, then the editors in high impact papers want this. So imaging is almost mandatory if the work, if you have such kind of life science work, and you can do it. And also these beautiful images, they are quite important for reviewers and editors, because if you have just lot of Western blots, it's good of course, but it's not convincing so well as nice, good image, which is also having numbers, so quantified image. So then back to this, is that imaging is very important. You can image nowadays a lot of things, not everything, but a lot of things. And most importantly, the imaging should be quantitative. Without that, it makes no sense. Okay, thank you. Do you have any questions regarding some methods or general questions regarding imaging? Or you have your own work where you are trying to use imaging? Other questions?
Mitochondrial aging, do you do this live cell imaging?
We are doing it with live cell imaging, but it's also possible to fix and to do it after fixation, image after fixation. But how we are doing, we are transfecting our neurons. We are waiting for maybe it was 4 days, and then we are doing imaging. We transfect in the probe, this mitochondria targeted mitotimer it is called. What people are also doing is that they are using the inducible mitotimer, so that they will transfect. They will express few days, and then they will add a tetracycline to block the generation of the new protein. In that case, they might get more precise data. But the variability in these experiments is huge, and it's not so easy as it may look. The variability is always an issue, because the signal is having different intensity in different cultures, neurons, and you should develop the assay which minimizes the variability. And also if you take these experiments, then some experiments are simple, but some experiments like this fusion and fission assay, they require extensive experience. So in our lab, I think people are learning it 3 months before they will get reliable data out. To measure mitochondrial cytosolic calcium is simple. You are just doing it a few times, and after that you can measure it properly. So something is more complicated, but it's always like that with all methods. It is like that. Any other questions? Sorry? Okay.
Mitochondrial complex, what is the function of complex mitochondrial complex 1 to 4 and
So to measure the activity, or what you exactly asked? What exactly you are asking? Sorry.
I'm asking about in mitochondria.
Yes.
5 complex.
Yes. Yes.
What is the? I am asking about what is the function of complex 1 to 4 and 5. I hear about that.
Okay. So what I got it now. So they are all, not all. So I think we should go. Okay, I will not go, but to explain it properly, you need to take this figure we had, or from the web, and it explains which complex are using NADH to produce electrodes, how electrons are moving through the complexes, and how the protons will be produced. So the main idea of these complexes is, of course, just to use this NADH to produce proton gradient. If your question was why we have so many, not just one complex, but 4 complexes to produce this, then I don't know. I think they are more efficient than one complex could be. They can get maximum out from this NADH. The role of complex 5 is ATP synthetase. This means that the protons will just go back from intermembrane space to matrix, and the power of this will be used to produce ATP from ADP. But if you want to know how exactly electrode, what specifically specific complex is doing, and how the electrons are moving, transported, then you should check this textbook biochemistry textbook figures. This is quite complicated. I don't remember which complex gives electrodes to another complex. I hope it helped. And just one more thing is that you have, at least in Europe, in US, you have a lot of people who are molecular biologists and cell biologists. And you don't have any, you have of course, but you don't have too many imaging specialists or the cell biologists who are very well educated in imaging. And at the same time, the imaging is developing very fast, and you need these imaging specialists everywhere. So we were looking for imaging, senior researcher in imaging, for imaging a few years ago. We offered quite good salary, and we were not able to find almost anybody. All applications we got, they were from cell biology, molecular biology, minimal experience with imaging. So that this is good to know about imaging, to show that you have experience in imaging in your future. It's, if you can choose that you have a core facility also, this university where you have a microscopes confocal, as I understood, if you can somehow use it for your work, then use it. It's good for your work, it's good for your CV. because animal experiments are good, cell biology good, but it's just my opinion that imaging is in the future. Maybe because I'm just an imaging person, but okay. Okay, thank you. Yes.
As we are advancing into imaging, I think one of the goals would be to see that in a lot of disorders, in a lot of diseases, these systems are compromised. So mitochondria are
Yes.
compromised, and as we move forward, either they could be, I don't know, they could be sort of targets to either repair, quote unquote, repair those problems and reverse that, or are we also, is the industry or the academia also looking at the synthetic form of mitochondria generation? Like, if that makes sense.
You mean synthetic mitochondria which will produce energy. It's how to say. At the moment, it is science fiction because you can't really produce ATP differently inside of cell. You can't produce or provide ATP. There maybe some 20 years ago, when I was PhD and postdoc, then a lot of people studied the phosphocreatine-creatine cycle. Do you know something about phosphocreatine? So that ATP energy in cell, because ATP is not diffusing so well in cytosol or not so quickly, then at least in heart and muscles, it will be converted to phosphocreatine, which diffuses quickly. And if phosphocreatine reaches to some destination, it will be converted back to ATP again. So it was the energy transfer, to transfer ATP energy. So people have tried to use this to give use phosphocreatine to give energy to body, to cells, but it's not really working for some, I don't know the reason. Phosphocreatine not going to cell, or it's not enough. So it means that I think at the moment there is no way to give such kind of energy to replace ATP in cells. And it's unlikely that there will be way. Of course, you can try to activate glycolysis to get more energy from glycolysis, but it's not working for the neuronal cells or cardiac cells, where you need quickly a lot of energy and glycolysis can't give it. You need mitochondria for that. So I think they are not replaceable in futures. So you have to study them next 100 years.
But technically, I mean, again, a lot of things have come into our realm from the creative ideologies of, when they were fiction at once.
Yes, of course, the social media, for example, was fiction 20 years ago. But some people, yesterday I spoke with one of you, and she told that they are studying the mitochondrial transfer. So that one option is that the mitochondria you have in test tube, that doesn't matter how you got these, that you will introduce back to the cell. But will it really help? Because in cell you have a lot of mitochondria, and if you will try to introduce some mitochondria, then you have so small amount. So you introduce 1 million mitochondria, but your brain is so big, so you will have 0.001 mitochondria per cell. It's not helping. So you can't deliver so much mitochondria as is necessary to get some improvement.
So right now, the more realistic thing would be to and
Yes.
sort of poke it to create more energy, if that makes sense, or either reduce the chances of mitochondria being destroyed.
Yeah, exactly. So that the main thing is to improve the mitochondrial health, to keep them more happy, to avoid mitochondrial stress and damage. And I was not talking at all about mitochondrial biogenesis, because it's so mitochondria are generated, there is biogenesis, and then there is mitophagy, they are removed. You can also activate mitochondrial biogenesis to get more mitochondria. It's PGC1 alpha activation. It was again very popular 10 years ago, but nowadays somehow people are not anymore very active in that field. But this PGC1 alpha is the master regulator of mitochondrial generation. Its activation could theoretically also help. People were looking for its activators very actively. They had no specific activators, but they had relatively unspecific ones, but somehow the results did not lead to anything important.
So, just a curiosity, is mitochondrial DNA also subjected to epigenetic effects, and what is the compatibility?
To be honest, I don't know. I think no, but I do not. I do not know that. Sorry. Okay. So if no more questions, then tomorrow I will also shortly speak directly about the science, what we are doing. Wolfram syndrome. How we are looking for Wolfram syndrome treatment targets. So we have some figures that you saw today, also tomorrow, but tomorrow I will explain why we did what. So okay. So thank you. I don't know. You have lunch probably now, or okay.
Okay. Okay, all set. Good afternoon, everyone. I'm Dr. Ashutosh Rai, and I did postdoc from UK as a Newton Fellow, and then I joined Department of Biochemistry as a Ramanujan Fellow. Today I'm going to talk about the topic of proteomic perspective on neurodegeneration, navigating molecular pathways in neurobiology. So, what are the neurodegenerative diseases? Yes?
So diseases in which there is a progressive loss of the main brain components. For example, there are neurons, or there are specific brain parts which are responsible for normal brain functions, such as hippocampus, prefrontal cortex, amygdala, etc. So there is a progressive loss or degeneration of those components, and these are called neurodegenerative.
Those components includes neuron or the some parts of the like glial cell, oligodendrocyte, all those things. So there are some genes established for some of the diseases, like for Alzheimer's. We know that there is amyloid. So amyloid beta plaque, or the overphosphorylation of tau and amyloid beta plaque accumulation. Then it includes a lot of diseases, like prion diseases are also part of neurodegeneration, Parkinson's. Then there are FTD, frontotemporal dementia, then ALS. So what is their hallmark of neurodegeneration? First thing is neuronal cell death or pathological protein aggregation, synaptic and neuronal network defects, aberrant proteosomesis, aberrant degradation of proteins, which is one of the most common example of this is, come on, APP. Then cytoskeletal abnormalities, altered energy hemostasis, DNA and RNA defects, inflammation, and all. So the first thing is how. So what are the proteins that get misfolded and result into the neurodegeneration, which include amyloid beta, tau, prion, alpha synuclein, and TBP43. So it's very easy. You can see there are monomers, then oligomers. So basically they accumulate and they form the fibril and ultimately the plaque. So based on the clinical features, neurodegenerative diseases are classified into two parts: cognitive predominant disorder and movement predominant disorders. So the cognitive, where the cognition, the mental part is involved, and the movement, which affect the muscles, so the movement is affecting. The cognitive part predominant includes the Alzheimer's, FTD, the frontotemporal dementia, the prion disease. The movement part include Parkinson's, atypical Parkinsonism, motor neuron, ataxia, and chorea. So we have already discussed the misfolded proteins are amyloid, tau, prion, and the location of the accumulation could be neuronal, astrocytic, and it could be oligodendroglial as well. So pathologically, Alzheimer disease is not, so we say Alzheimer disease as the mix. It includes both APP and the tau. So it's mixed pathology. So what happens initially, there are diffuse plaques of APP. Then as they get the progress, they get denser in the center and diffuse on the sides. This denser part further attract the tau accumulation. This is histologically, and then this tau accumulation, because of the overphosphorylation, it leads to it histologically it looks like accumulation of the tau in the neuron bodies. Then this was because of amyloid beta and tau accumulation, so mixed. Now there another protein which leads to NDD is alpha synuclein, and it includes Parkinson's and multiple system atrophies. So typical feature of Parkinson is Lewy body, and it could accumulate either in the so this is the immunohistochemistry of the protein alpha synuclein, and this is the pictorial cartoon so how typically it could be accumulated in the cytoplasmic part or in the fibrous of the neurons, and in the multiple system atrophy, in the glial, it could be either arranged in the sickle shape around the nucleus, or it could be in the nuclear. So this is the nucleus of the neuron, and this brown with the blue arrow is the inclusion body. Then these are these are various diseases leading to the neurodegeneration, and 3R, it could be because of the 3R or 4R tau. What is 3R and 4R? It's easy. What is 3R? 3 repeats and 4 repeats of the tau. So in case of Pick's disease, there is a spherical inclusion of the tau around the nucleus. In case of you can see that it is the tau which is getting accumulated, correct? But depending upon the location, and so basically depending upon the location and the number of tangles, it is causing a spectrum of disease. So what could be general symptoms of the neurodegenerative diseases, which is memory loss, motor deficit because of the stiffness, tremors, and dementia and impaired language. So far we find that okay, there is Alzheimer's by the amyloid beta and tau, and alpha synuclein in Parkinson's, then 3R tau R spectrum. Then, but it is not that Alzheimer, the neurodegenerative diseases research has stopped, that we have done everything sorted. No. Why? Very good. Before that,
they take very long time to.
very good too. And one of the very common and most important reason. Yes, that's the future. Yes, the common heterogeneity. So yes. So sometimes, so they take, one thing is they take asymptomatic when the first symptom appear, and when it is the full blown Alzheimer's, it sometimes in some people the duration is very long. So first thing, why there are considerable, I will say, markers, but why we still need to find a good reliable biomarker both reason accurate and reliable diagnosis, prognosis mainly, and the as you mentioned, the therapeutics. So first, many neurodegenerative conditions have extended preclinical and prodromal period where diagnosis using available symptom based assessment is either not possible or extremely difficult due to the subtle manifestation. Second, yes, the heterogeneity because of the co-pathology and multiple other diseases along with the neurodegeneration. So, what could be the most easy way to find a biomarker and most reliable? So, we can look for the DNA for the genetic marker, we can look for the RNA by transcriptomics, and we can look for the proteins, but then which you will choose and why? Sorry, proteins why?
If you have a DNA based or RNA based marker,
Yes.
so the process that will have to go through in order to identify that marker in a suspected patient would be comparatively, I would say, complicated.
No, I will not say. RNA, yes, DNA, no, but that's very complicated explanation. Let's find out the easy one. What is the central dogma? DNA, RNA, protein. So who is the ultimate functional machinery? DNA is not causing the tangling, DNA is not phosphorylating the tau. Who is the culprit? The proteins. I will say the functional machinery. Ultimately the thing is done by proteins. Yes, DNA is very good because it's predestined. Okay, DNA, so predisposition. So we know that there is some mutation in APP and some genes which lead to the accumulation and that ultimately lead to the tau accumulation and everything. Yes, but in quest for the ultimate functioning person, which is protein, we will look for protein as a biomarker because it's easy to, so we target proteins. You do not target DNA, we target the kinase inhibitors, the trametinib, sunitinib, nilotinib, the kinase inhibitor, senescence inhibitor, the quercetin, dasatinib, all those things. So we target the proteins. So if we found a good biomarker, it will be easy for the, I usually not say diagnosis, I say prognosis. So a good protein biomarker will help in the prognosis and the therapeutic. So we are moving towards the proteomics. What is proteomics? Study of? Very good. Yeah, proteome is like 30,000 proteins. So study of the large scale of protein, large number of proteins, and there are multiple ways. So you could do the qualitative proteomics and you can do the quantitative proteomics. What is difference between qualitative and quantitative?
The identification of a particular protein, and quantitative would definitely be the amount of quantity of.
Yes. So she is right. If I do the qualitative proteomics, and I am comparing Alzheimer's versus the healthy person, I can only find out this protein is present and this protein is absent. So but it may not be possible. So but if I do the quantitative proteomics, I can also tell that this protein is, then I can compare and I can tell, okay, this protein is 2 times more in Alzheimer's compared to the normal healthy brain, or 3 times less in Alzheimer compared to the normal healthy brain. So or you can and that part of information of the qualitative will definitely be in the quantitative. If a protein is totally absent in normal but present in the Alzheimer, it will appear, it will give its value, the numerical value in the Alzheimer part, but there will be nothing in the normal healthy part. So the qualitative will give you the protein profiling. So it's profiling. A, B, C, D is present in the Alzheimer and A, B and C is present in the normal, so D is absent. Just profiling. And when I will say that, okay, A is 2 times more in Alzheimer's compared to the normal, it becomes quantitative. And what else you can do in qualitative proteomics, you can characterize the post-translational modifications. But usually we also do the quantification of post-translational modification, and the third interesting part you can do with the qualitative is analysis of protein-protein interaction. Any guess how?
It can be like an receptor.
Very good. I will say using the affinity chromatography followed by affinity chromatography then mass spectrometry. This is what you, yeah. So it's like but always it's not ligand receptor. If there is a protein and its interacting partners, if you want to find out interacting partner of, let's say, HSP27, hitch of protein 27, so you will precipitate the hitch shock 27, just normal affinity immunoprecipitation, very simple No, you are not happy?
What he said would be effective in case if the other protein that is binding to the target protein would be covering the epitope, which is supposed to be bound to the antibody. In that case, maybe the antibody can be hindered from binding to that protein.
It depends what whose KD value is more or less. Then it becomes proper chemistry. But just imagine, here is my mass spec data, here is the in silico, what does it match? So if you are not happy, okay, so moving and quantitative, we get the numerical information. Quantitative proteomics, using quantitative proteomics, what you can find, you can do the molecular mechanism or signaling pathways, you can find out the disease biomarkers or identification of therapeutic targets. How? Let's go one by one. The second point is clear, correct? So you can by doing the quantitative proteomics, you can find out a biomarker or let's say prognostic marker or diagnostic marker of a disease, something. So Alzheimer's is going to be symptomatic, early onset versus late onset, who is going to be symptomatic, who is not going to be symptomatic, all this you can do. Then what is problem with the first part? You will get a list of 20,000 proteins and then you have to map it depending upon your particular interest. So if let's say out of 20,000, 500 proteins are significantly upregulated in symptomatic patients, then your further concern is only that 500 proteins, not the 20,000. Don't get attached to the data. So those 500 proteins, and then you will put that 500, you will do the bioinformatics on those 500 proteins to find out in which molecular pathways they are involved. So you will map the signaling pathways, and then you can say that okay, 30 proteins are up in particular XZ pathway, and this is what's supposed to be. So I say that okay, EGFR gives the signal EGFR, MAC, ERK, and then further downstream. So if what in my data EGFR is up, MAC is up, ERK is up, and HSP27 is up. So this is what theory says, this is what my data says. So why not I will choose this, and I will say okay, this is the pathway of my disease. If okay, and depend, so you always get the significance p-value, and then if you identified the you have already identified the proteins, you map the signaling pathway, then you can target those proteins. Correct? You have not heard the mTOR inhibitors, you have not heard the integrin antibodies for integrin is everolimus, bevacizumab, which is VEGF inhibitors. So yes, if you identify a protein, you are close to identifying the therapeutic target. But how you do the quantitative proteomics? So you can do there are by two ways, labeling and label free. So label free is it's, I will not say easy, it's cost effective. You just count the spectra for each sample, for each protein, not you count, the software will count, but it takes time, and measure the ion intensity and the spectral counting, and then you combine both, and then you say that okay, this is up compared to this, and you have to do that independently for both the control group and the disease in question. On the other side, the labeling is easy, more reliable, faster. So then you can do labeling by two methods. There are two ways, in vivo and in vitro. If you are doing like culture, and your research question is treatment versus not treatment, you have a model of Alzheimer's disease, you have a model of some any disease of your interest, then you can have that model versus control. Let's say we know that if there is mutation in APP, it can lead to the Alzheimer's. So we knock out or we induce that mutation in a cell, and so now that become the Alzheimer's model, and then we have the wild type where there is the normal copy of APP is there. So then you culture it, then you use you instead of then in the culture media, our media is normal, while when we do the in vivo labeling, we use heavy and light isotopes, and usually on the lysine, and so N14 and 15. So one we will give to the wild type model, and another we will use to the knockout model, and we will subject to the mass spec, and when the the software will compare itself, it will know that okay, this carbon is heavy isotope, this carbon is light isotope. and this method was discovered by my boss's boss. Then comes the in vitro. In vitro method is more handy when you are doing the tissue proteomics, the blood, plasma, all things. So then you isolate the protein, you do the trypsin digestion, and then you label, again the thing is same, you label those peptides with isotopes. So and then isotopes, they don't get like ITRAC, TMT, tandem mass tag, these are like, just consider them as the barcode. Okay, so let's say we have two groups, Alzheimer's and control, healthy. So I will label the Alzheimer's with barcode A, and the healthy control will be barcode B, and then whenever in the mass spec it will know that okay, this peptide is barcode A, this peptide is barcode B, but they will be close, correct? Because mass spectrometry, it is the basis of mass is to charge ratio. Those two peptides which are same size but from different samples will be close enough, they will be close. So but the intensity may be different or may not be, but the mass spec will identify, okay, the A is more or B is less, something, they will be close, and it will measure the difference in the intensity. So what samples you can use in case of neurodegenerative diseases? The cerebrospinal fluid, the CSF, which should be the most ideal sample because it reflects the brain thing, okay, it reflects the brain pathology, but it's invasive, I will say it's painful. Anybody has seen the CSF sampling, human CSF sampling? It's painful. So they take from the lumbar spine and with the needle. Second easiest thing is blood, like plasma or serum. It gives us the information, but it is not exact reflecting the brain pathology. But everything has its pros and cons, so it's less invasive than the CSF. Then the easy one, the saliva, tears, urine, okay, everything is happening, so maybe something correlate with the saliva, tears, urine. Now apart from liquid biopsy, you can do the proteomics with the tissue sample. How you will get the Alzheimer, let's say Alzheimer patient, human with Alzheimer's, how you will get the sample? Very good. There is no other way. No other way. So the autopsy samples. So whenever, if you will read the paper, I found most of the time autopsy sample only, and nobody is going to, they suffer Alzheimer, Parkinson, I am not going to give you the permission to make a hole in my brain and take one part of the sample. No, sorry, nobody gives. So the only option is autopsy. So you get the brain, then one thing is, next slide, sorry, the whole proteomics, the bulk. You get the affected part, the pathologist will tell you this is the affected part. So the pathologist will give you the sample, you take the sample, process it. but that is called bulk proteomic. If you are rich enough, if you are a rich lab, you want to get more fancy and you want to be the cell specific, so then you can do sorting, micro manipulation, labeling, imaging. So sorting, it's very easy. You make the single cell suspension of your tissue, the tissue, make it single cell suspension, and depending upon which cell proteomics you want to study, you need to know the marker. The only drawback of the FACS is you need to know the marker. So which type of cell you want to do the subject for proteomics, you sort it, and then after that is the same, subject it for the mass spec. So the good part is highly pure. It gives you the real answer, but the bad part is cell damage during the processing. There are a lot of processing steps. The microfluidics is again the same thing. It's like these days they are very thin capillary kind of thing, and they subject the cells to you have to make a single cell suspension, and they subject it to that fluidic system, and it allows only one cell at a time to pass, so you will get the single cell suspension. Then the most interesting thing is mass cytometry. Anybody has heard about mass cytometry? So mass cytometry is like FACS. It's like FACS, but in that case we do not use fluorescent labeled antibody. We use metal tagged antibodies, and that's it. This is the only difference between mass cytometry and flow cytometry. Then the manipulation, micro manipulation, it's easy. Laser capture micro dissection. You decided that you want the core of that plaque. You did not trust the literature. You say everybody is saying that there is a plaque. What if there is something more? So you want to take sample of plaque, and your research question is plaque versus no plaque. So you take sample of the dense core and go slowly, like every 1 millimeter from the core, from 1 mm from core, and then 1 mm for core, and you compare all the three samples, and that you can do by either by only if you have the facility of laser capture micro dissection. So in case of laser capture, you get the sample on a slide which has the PET membrane, the normal glass slide, but that will be the membrane, and on the computer we have the image. We say we want this part, automatically the needle will go and take the sample from that part. Another thing is micropipette aspiration, that anybody, we all do. I say that macro dissection, because if we are not rich enough but still we have the sample, so we go to the pathologist, we say please mark the core versus not core, and then we take that part with the fine needle or the tiniest pipette tip. The another thing is labeling. So we label with the particular enzymes, like or proximity labeling, like we label that, okay, this is, now we take the sample, make it single cell suspension, we label that particular cells, and then subject for the mass spec. Then the last thing is mass spatial, it's spatial proteomics. I do not say proteomics because till date we are not doing full proteomics on the spatial. So, but still people say it's proteomics because we are moving towards. So what is spatial proteomics? Have you ever heard of spatial transcriptomics? No, no. It happens on the slide, the histology slide.
Specific location those evaluated heterogeneity.
So when we get you process the bulk tissue, you got the proteomic data, but you do not know that which protein is coming from which cell. Perfect. Then you did the single cell suspension, you separated the cell and you did the proteomics, you got the information of proteins from the cell, but you don't know where this cell was histologically, what was the location, correct? But the spatial proteomics is giving us the advantage of it does on the slide. So from each cell it gives us a colored photo. It from each cell it will give us it will say, okay, this is oligodendrocyte and this is the proteome of oligodendrocyte. So it will tell me, okay, you will get very fancy colorful image of the slide. Usually what we get from the histology slide, either brown color, brown and blue, the IHC, or 3, 4, 5, maximum 6 to 8 colors on the, or if we are doing multiplexing of IHC, 6 or 8 colors of the immunofluorescence, sometimes 40 if someone is going to be more picky, but the spatial transcriptomics give you each cell type. So it will say, this is the endothelial cell and is this the proteome, and there are 20 endothelial cells, there are 400 oligodendron, there are 400 Purkinje fibers, and this is the proteomics, and it will be colored. So it gives pseudo color to the each cell, so you get beautiful color map and parallelly a data. So you got the sample, and based on your research question and I say affordability, what technique you can follow. Now what proteomics you are going to do? The whole proteomics is two things, the bulk thing and then just proteome. You process it and you get it. But what is the PTM? The post-translational modification. So it is not necessary before that. How many type of post-translational modifications are there? Name 2, 3. Methylation, phosphorylation, glycosylation, ubiquitination and acetylation, simulation, blah blah blah. List is long. But phosphorylation is the most common one. And it is not necessary. So if you find a kinase which is overexpressed, so what is requirement of kinase? Or it has to be active. So kinase, so let's say I am in the department, I am teacher, but I am not teaching. Okay, I am not teaching. So what I am doing? Nothing. So I am there or I am not there, it does not matter. So that's the position of a kinase. So if a kinase is overexpressed, let's say there are 20 copies but not active. So usually it is not always expression that matters, it is the activation also that matters. So once it gets phosphorylated, it gets active, correct? Agree? Everybody agree? Nobody disagree? Not always, the site specific. Very good. Then who will give you this information? The mass spec, the proteomics. So you can do again. So let's say the phosphorylation. We want to study the phosphorylation, the most common one. And phosphorylation happens on how many sites? Where? What are the sites? Sorry? Very good. Serine. Someone said something. Threonine. And the most least abundant but most important? Yes, tyrosine. And these days they have discovered that histidine also get phosphorylate. But let's ignore histidine. So tyrosine, serine and threonine, these are the most commonly phosphorylated. And yes, you are right, depending upon the site it gets activated or deactivated or inhibited. So how we get the phosphorylation? So when we do the proteomics of phosphorylated peptides, we say that phosphoproteomics. Okay, so phosphoproteomic, glycoproteomics, that's it. I have never heard sumoproteomics. So but then ubiquitome and acetylome, so they leave the proteomic part and keep the ome part. So phosphoproteomic, glycoproteomic. So how you get the phosphorylated peptides? You got the proteins, you got the sample, extracted the protein, and now you have extracted a protein, digested with the trypsin. Now what? You have 20,000 peptides, not everything is phosphorylated. So, and I want only phosphopeptides with phosphorylation. Yes, yes, and then what? Yes, you are right. Yes, go further in the right direction. No, we don't do ion exchange. Ion exchange is the part of mass spec, but we don't do that for enrichment. Okay. Yes, affinity. Yes. So how? Yes, she is 70% right. I am very happy. So because the peptide which are phosphorylated has the 2 negative charge, so there are multiple affinity chromatography methods to do the enrichment of phosphopeptides. So you can do with any metal which has 2 positive charge. So nickel, Ni 2 positive, Fi 2, Fe 2 positive, iron, or IMEC. Someone presented the thing IMEC, immobilized metal affinity chromatography. So then initially people say, okay, nickel 2 positive, very good, we found the method. So they did not get the enough number of phosphopeptides. Then someone says, okay, Fe2 positive, let's do Fe2 positive. They did not get enough number. Then someone combined the metals, Ni, nickel and Fe2 positive both. So instead of saying metal, so they immobilized metal affinity chromatography and they get better results. What I do is titanium dioxide method, so TiO2 positive. So then titanium dioxide, it's easy, it's very easy. You have to get the beads, incubate your peptides with the bead, elute it, done, sorted. So and this process is called enrichment. So we do the phosphopeptide enrichment, we do the glycopeptide enrichment. So when we take out something exclusive which is of less stoichiometry from the mass thing, we say enrichment. So this is called phosphopeptide enrichment using your choice, nickel, imac, titanium dioxide. But the drawback is you get very less number of phosphotyrosine, because the most abundant is serine. The ratio is 92 to 8 to 0.1 in normal condition. So tyrosine is 1 to 0.1% in normal physiological condition. So we do not get phosphotyrosine enough to draw some conclusion. Then what we do? Is there any way we can do something? Then we do the immunoprecipitation. Now we know that we want the phosphotyrosine thing, so we get the antibody which detect the tyrosine. So we get the anti-tyrosine antibody, and in the world there is only one antibody that everybody use. So the phosphotyrosine antibody, and do the immunoprecipitation, and you will be surprised to see that you will not believe that, okay, this much number of tyrosine were phosphorylated in my sample. So yes, enrichment, immunoprecipitation if the stoichiometry is further low. Then, so that was the whole proteomics. Then the modifications. Then is the aggregated, if you want to study the plaque, the infarction, the previous, you just need to take that part and subject it to the mass spectrometry. Spatial we discussed. And how do you study the turnover? I just told. So proteins get degraded when they get no, not because of us. They get degraded on the temperature problem when we do physiologically. What is the modification that guide the cell that, okay, this guy is ready to die? No, ubiquitination. Don't talk miRNA to me. I'm not.
So what is the name of the antibody, anti-tyrosine antibody?
PTYR 1000. So it's like phosphotyrosine 1000. It's very costly, super duper costly. But yes, if you want, you can do. But it's worth trying. If you think of tyrosine phosphoproteome, it's worth trying. If anybody wants, I have it. At least I'm rich on something. So briefly, you do the proteomics, you find out the disease biomarker, and by mapping the proteins to the pathway, you find out the mechanism, and then you can target. And also, why the first problem we discuss is the heterogeneity. So my proteome could be different from the another person's proteome. So because definitely there is a reason why one person is developing at 20, why one person is developing the symptoms at 40, another at 60, and so there is definitely difference in the proteome. So that's where comes the role of personalized medicine. So if you, this is very, we all say this, we all are trying this, but this is very dream situation, that whenever we get the sample, it's like for the personalized medicine, things should be happening in the pathology, correct? Sample ko, they perform mass spectrometry on every sample and they say, okay, this pathway is in this patient, this pathway is in this patient, and then comes the problem of clinician. Okay, he has to read every report and say, okay, you are going to get this, you are going to get this, you are going to get this. But this is our dream. We are working for the humanity. Back to the phosphorylation. What are the functions that are regulated by phosphorylation? It's not just activation of kinases. It is involved in the molecular association, enzyme activities, intracellular localization. Agree? It's very important one. And then crosstalk, when something get phosphorylated, then something get acetylated, or if something is acetylated, it does not get phosphorylated. So the crosstalk, protein degradation, when something is phosphorylated, then it gets deactivated and it does not lead XYZ protein to get ubiquitinated, so the protein is not degraded. So it controls the protein degradation, protein stabilization, conformation, and processing and receptor cycling. And the processes regulated by phosphorylation is, there are vast functions including neural plasticity, cell growth and adhesion, differentiation, everything. Very briefly, this is what I do. So you get the sample, just omit my part. So and you can have any sample, your Alzheimer's, normal brain, you take the protein extraction with the help of lysis buffer, and then we do the trypsin digestion, because we want the we in the mass spec we identify the peptides, and then we label these peptides with the tags in an easy language, the barcode, and then we pool it. So this is one of the advantage of labeling. So I started with 3 samples and I end up with 1. So from this step onward, I have 1 sample to process, but and the cost is this thing, the TMT tag. This is the costly affair, but then after that it makes the life easy. So if you are doing the enrichment or even the mass spec, if you are doing normal proteomics, go directly from here to here. The sample is ready, labeled, subject it to the mass spec. One tube is going for the mass spec. Imagine you were doing the label free, so you have 3 samples will be going, so the cost will be 3 times, and it will take more times to process, it will take more time to analyze, and also if you have planned something, the PTM study, you have to do enrichment, now you have to do the 3 enrichment in 3 tubes. And what if your antibody is PTYR 1000, you have to take 3 aliquots. So everything has its pros and cons. It's costly, not very costly, it's costly, but then it makes life easy. After this, 1 tube enrichment, 1 tube subject to mass spec. Mass spec is easy peasy, it will give you the quantification, and this is called relative quantification, and then that's it. This was the process. this I bring for you. So our dear spatial proteomics has been said as the method of year 2024. So in the nature methods, so this is the fancy thing, and everybody is doing spatial, whatever they can do, spatial transcriptomics, spatial genomics, spatial proteomics. Everybody, now we don't want the proteins in the tube, we want to identify protein in the tissue itself. So you can see, so they have labeled hippocampus. So this, the colorful thing is hippocampus, and they of Alzheimer's. I struggle to find out this good one thing, and then different cell types. So neurons are orange. Where is orange? Orange, magenta, green, astrocyte, everything. But the interesting thing is this one, the plaque. So they manage to color everything. Then they have the data. So they have the data of this purple the proteomics data from the purple thing, proteomics data from this orange thing, proteomics data from the yellow thing. So now they know this is the arrangement. Plaque is here, which is between astrocyte and which they say magenta thing is neuron. So between neuron and the astrocytes are the plaques, okay. But this information you will not get if you crush the whole tissue, correct? And it's always, nature is more smart than us. We have reached to this close, but we say that okay, here is plaque, but who decided the plaque will be here? There must be something, and that something information we are getting closer to and closer. So that something information can come only by spatial transcriptomics, because this will tell me that okay, these endothelial cells, these astrocytes, when they get changes in XY protein, they lead to the accumulation of plaques. Then another fancy thing is single cell proteomics. You have heard of single cell transcriptomics, single cell genomics? No? So it's okay, everything is same. They have just, instead of doing the bulk, we are doing with the single cell. And doing the single cell, there are multiple ways. So one thing is you can do the FACS, but nobody like it. The fluidic method, the microfluidic is yes. So we get the machine which has the plates with the microfluid, and it allows only one cell in each well. So yes, this one. So this is the, you will be having, let's say 96 well, and each well will have further wells, and the well will contain, each micro well will contain only one cell, and then each micro well will have its own barcode. After that, barcoding is done, so each cell is coded. Now I know that cell 1 is whatever is it is, cell 1, cell 111, cell 2, cell 2.3. So every cell is coded. Then after that it's same. We perform the proteomics, and then we know based on the biomarker for that particular cell, okay, this proteomic signature is coming from the glial cell, this proteomic signature is coming from the astrocyte, and that's it. Now to change the flavor, the real data. How many people are working on neurodegenerative diseases? I know, except me, most of you. Or everybody is working? All people are Alzheimer, Parkinson's. Very good. Are you aware of this consortium? No. So they made a global, there is a global neurodegenerative proteomic consortium. I know, I am not the, so this is a part of, are you aware of Human Proteome Atlas, Human Protein Atlas, HPA? No. HUPO, Human Proteome Organization. You know HUPO? Okay, so there is Human Proteome Organization, and which have centers across the world, and in India there is only one center in Mumbai, Dr. Sanjay Navani. They are pathologists, and or anybody, one thing they are designating, anybody, I can submit my IHC or proteome data, that here is data, you use it for your thing. So they made the Human Protein Atlas, so that contains all the protein information, the real information, the immunohistochemistry. Now they have made the cell atlas, this. So finally the same people have made the global neurodegenerative proteomic consortium. So this include specifically Alzheimer's, Parkinson's, and the lateral sclerosis, and this dementia, frontotemporal dementia, and aging. And this is from the 23 cohort, means 23 centers across the world, and look at the sample size, 31,083 samples they have processed. And I'm getting too much time. Please, so and then for these samples we will go on till someone comes. So this is interesting. So they have performed, so in short they have large cohort of sample representing the whole world. So they have sampled definitely from Asia. I have not read there India is there or not, but Asia is there, Asia, UK, Europe, everybody. And then they measured 250 million proteins, 250 millions. And what they have found, so everybody is familiar with this, the volcano plot thing. And so they compare Alzheimer's versus normal control, Parkinson's with control, and what they, so these are the upregulated proteins, these are the downregulated proteins, and in the same case. So if you are aware, you know better than me the neurodegeneration. Alzheimer's, it is acetylcholinesterase, and so these are the list of genes which are up, and same case upregulated in Parkinson's, and interestingly there is nothing upregulated in frontotemporal dementia, it's downregulated, and yes, in lateral sclerosis. Then you perform the mass spectrometry, you decided to work on neurodegeneration, you got the tissue cell, whatever you got, you perform the mass spec, you got the data, you converted data into this fold change thing. Now you know that okay, these proteins are up, these are down. What next? What you can do with these? This is not enough until unless you say that okay, this protein is involved in this process via this pathway. So you have to find out the molecular function, the pathway analysis, and protein-protein interaction, and many more things. But these are the main thing. How you do this? So there are two methods. One thing is paid softwares, and another thing is freely available. So anybody aware of this? Ingenuity pathway analysis. So this is paid software. It's from GIAN and it's paid software. It gives the free trial option if you have the institutional ID. So, but it gives you very good information. It can analyze your upstream regulators, signaling pathways, and it can tell you that your protein is involved in which other diseases, so you can correlate the pathology, and it can compare the expression pattern, mechanism, everything. And this is the real data, this is my data. So, and it is and it can, so the red thing are overexpressed and green thing are underexpressed. So it will map what is the location as well. So I know that these protein are membranous, these are cytoplasmic, up and down, what is up of this, what is down of this. So I can think about the mechanism. But that's not the end of the world. If something is paid, there are free software. This is totally free. You can enjoy. You have to download this. So the fun rich. I love this software. It gives plenty of information. It gives you the gene ontology, biological pathway, protein domain, site of expression. Like it will tell you ki, okay, you found this protein in Alzheimer's, that's your data, but the software will tell you, okay, this could be the microglial cell, this could be the astrocyte, this could be the lymphocyte. So they give you a number and the p-value. So we choose the first criteria in biology is the p-value. Something has to be significant. So based on significance we select, and it gives you the transcription factor. So difference between this one and the free one, software does for you. You say click, just click the button and it will say, okay, I want the upstream upregulator of the TGF1. So from your data it will do this and it will give you beautiful diagram like this. Here look, it is giving all the information. It will tell you the pathway, it will tell you the transcription factor. So if you know the transcription factor, if there are 10 proteins which are overexpressed and you know the transcription factor, so you definitely know that this is the transcription factor, it is leading to the overexpression of these genes. And then you can go back to the literature that this transcription factor get activated when there is activation of, for example, NF-kappa be activated when GSK3 beta is phosphorylated, correct? So when it's phosphorylated, NF-kappa B is activated, it goes to the nucleus and do the transcription. So you can go, you have to go back and forth and you have to use your time. I will not say waste, but use your time and the intellect. I love this. Then there is, this is very easy peasy tool. It's ShinyGo. It's online web version. You have to do nothing. Just paste your list here and it will give you so much information. It will give you the pathway involved, and you people have heard of KEGG pathway mapping. You need not to do this. This guy will do for you. So it will give you the pathway, this, and then you select the pathway which one you want to map. So it will give you, okay, this is the pathway, and it will do the color thing. Okay, in this pathway, let's say P53 pathway come up in your thing, and all the red one are upregulated and they are in your, so out of 20, 10 are in your data. Then usually we do the string. Have you ever heard of string? So we do the string analysis for protein-protein interaction. So this software even cut that. Okay, don't go anywhere. If it's like mall, if you go to the ShinyGo mall, everything you can do in the same place. So you can do find the pathway, you can do the mapping of the pathway, you can do the protein-protein interaction, and which I love most interesting is it gives the genomic localization. So you can go back to the gene. So now what this information gives, that, so this is the enriched, means whatever this particular, so this figure says that chromosome 20 this overexpressed. So mostly overexpressed gene is on chromosome 21. If we zoom more zoom, it gives exact location. Then you can, then depending upon your research question, you can go back and say, okay, I want to do the gene amplification, like I want to do the PCR, that what if there is copy number variation. So what if the problem is not at the transcription and translational level, but it's at the genetic level, there is duplication of this part, correct? So it gives you the genome location also. Back to the continuity of this data, which is your Alzheimer's thing. So based on all those, the pathway analysis, they did the, after doing the volcano plot and expression, they did the pathway analysis, and they find out that in Alzheimer's, this is the glucose metabolism and regeneration and endosomal sorting, these pathways are up, while in Parkinson's contains a lot of GTPases. So it's full of Rho GTPases pathways, and the temporal dementia has protein translational, protein phosphorylation, IGF signaling, and so doing all those mechanisms which I explained, they mapped. Don't worry, this is just a different way of presenting the data. We say this is the bubble map, so you can do by whichever. Another interesting thing you can do with your proteomic data, so they have predicted the difference between the chronological age and the predicted age. So based on proteomics, they correlated that based on this proteomic, what is the does the proteomic age correlate with the chronicle age, and they found that yes, it correlates, and using this as a baseline data, this was the baseline, this was the previous mechanism. What is the information that usually, what do we think? Alzheimer's brain is the problem, but they have shown that cognitive brain mostly correlated. So there is in Alzheimer's, the gap between the chronological and the disease age is more in the cognitive brain, the bold ones, the artery, the liver and intestine. I have not imagined about this information. And then in case of this dementia, it's cognition brain and artery. Then in the Parkinson's, muscle is expected, but lungs, maybe because ultimately it has to function in a muscular way, but Alzheimer's was totally surprised for me. So proteomics can give you the pathway mechanism, it can tell you. So what did information we get from this? That nobody knows that this lead to this or this lead to this. So the paper quote that we don't know its causation or consequence. So you have the arena open. So another study they did the plasma proteomics of, so this study is comparing symptomatic and asymptomatic patients, and they found yes, ACHA is there, but SPC25. And when they correlated with the clinical parameter, these number of proteins correlated. The red one are over, and the more positive correlated, and the blue one are negatively correlated. The interesting thing is here. So these 20 proteins, the MIA and called 10A1, these two proteins are involved in cytoskeletal and extracellular matrix, and they correlated with the symptomatic progression of the disease. So if there is progress, if the with progression they decrease. And then they correlated, they find out the pathway. Nervous system we expected, but the ECM and the lipid pathway may be new for this. Then what do you, so it's the neurodegenerative diseases, correct? So what they are having cognition affected, memory affected, so must be something which is common. So this study find out that 836 protein are exclusively to Alzheimer's and 673 are exclusive to Parkinson's and only 270 are for this dementia frontotemporal, while these are the overlapping ones. So between Parkinson's and Alzheimer's there are 1600 proteins overlapping, and between all three there are 996 proteins overlapping. Then you can go back and you can do the pathway mapping. You put as a homework, you can do as a fun, you go to this paper, take these 996 proteins or 16 and put in the shiny go and see the magic. Then you will find out that okay, this pathway is common to everyone and this pathway is specific. And thus this is the mapping like common and the specific one. And the last study, this is my favorite study. So they have done both proteomics and phosphoproteomics on the same samples, and this is like principal component analysis. They say that okay, this is like based on the age. Now people started correlating that okay, with age memory decreases, with Alzheimer's memory decreases. So what if we found the gene which is like with the aging, and what is common between aging and Alzheimer's? So if we found something for the aging, we can find the, can we target the same thing in Alzheimer's? So based on this question, they divided the animals. So this is animal study, everything else was human. So adult, mid-age which is green, and the old one, and you will find out. So this is the proteomic data, and these are the clusters. They clustered like up down on the, and you will see that these clusters, the proteins are overexpressed in old, and these is like, so only one case there is under expression. And what is, and this is the pathway. So signaling to the GTPase goes down as with the aging. So we can hypothesize that maybe this is something happening similar in the Alzheimer's. And what else is happening? Yeah, what is going up? Neuroinflammation. So inflammation, all this goes up with the aging. So we can hypothesize that maybe something like this is happening. Then based on this beautiful thing is kinome tree, and it represent, every branch represent the kinases, human kinome. These are 500s. So based on phosphoproteomic data, because you got the phosphopeptides, you go back to the in silico analysis, and you can deduce that okay, this phosphopeptide is phosphorylated by the kinase, this kinase. So from that information you can find out the kinases which are involved in your disease question. So here they have said that these kinases are involved in the aging. This is total they identified. And this is the phosphoproteomic data, and we can see that the overexpressed one are the yellow one and the old one, so the gray. We have to look at this, the gray thing, and gray thing is glutamatergic synapse, synaptic, So all this we found in the Alzheimer's. Agree? So we can correlate that something which is happening with the aging may be similar is happening in the Alzheimer's. Yes, they are definitely more smart than me. So they find out the phosphorylated, so they know, we know that these phosphopeptides are correlated with neurodegeneration, and these phosphosites are cellular senescence. The easy peasy one is the tau, MAPT is tau, correct? And all tau is hyperphosphorylated in the old mice. So now do you agree that with aging tau is getting phosphorylated, and that we know that with aging memory goes down, so maybe is a good model for the Alzheimer's, or at least it can generate a good hypothesis. So this is the kinome of aging. These are the kinases which are associated with aging. In 2 words, what information you get, that most of the kinases are from the CAMK family or AGC family or STE family. So you cannot target tyrosine kinase inhibitor. So nilotinib, sunitinib, dasatinib, blah 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 blah, any IB is not going to work on the Alzheimer's patient. So you have to find out something from the CAMK. You either the inhibitor is present, you can try. If it is not, you have to develop. So we know that the CAMK, or at least you get the filtered information that these are the kinases I have to find out, and this is what I do. And the same study showed that the kinome changes are involved in the neuroinflammation by cytokine signaling, neuroinflammation, and the clustering CD molecules. This is what we are doing with the Rajat sir. So he is focusing on the neuroinflammation part of the Alzheimer's. This study shows that with aging there is breach of the blood-brain barrier, and that can lead to the cognitive impairment. That can lead to the cognitive impairment, and the one of the most common. So how they say the breach is there, because they compare the CSF to the plasma ratio of the protein. So they did the CSF proteomics, they did the plasma proteomics, then they find out that which proteins have higher ratio. The CSF to the plasma ratio was high, and fibrinogen is one of the protein which correlate with the aging and the cognition. With severity of cognition, it's not very high, but it's high. MMSE is the test for cognition. This is the study. It's like ataxia related. It's the neurodegeneration. In this disease, the Purkinje fiber neurons, they get degraded. One of my colleague in UK have done this work. So and they know that this gene when it get mutated, it leads to the so look in the wild type. The green thing is vimentin. So in wild type, when SACS gene is normal, the fibers are nicely arranged, and when it get mutated, it is like there is no arrangement, it's accumulation. So that leads to the movement disorder. And when they did the proteomics and the phosphoproteomics, they found overexpression of tau and overphosphorylation of tau. So one more thing added to the tauopathies. And when they did the kinome mapping, these are the kinases they have given to, like the red one is overactivated, and the specific tyrosine 262 was, so because when they did the kinase enrichment, they find out that BRSK2 kinase. Nobody ever heard of BRSK. We nobody bothered to BRSK2, but this is the most significantly enriched. So when they validated, If someone will ask me to do the kinase, I will choose very fancy one, even the EGFR, AKT, ERK, MACPAC, but nobody goes to these one, and they surprisingly they found the knockout cells have overexpressed BRSK2 and hyperphosphorylation of tau 262. And then they did the surface proteome. So far you did the bulk proteome, you did the single cell level proteome, now you can go to the subcellular proteome. So this, when I say surface one, this is the membrane. So there are two ways to doing the membrane proteome. From the bulk, using the bioinformatics, you choose the only pick the membrane ones which are bioinformatically localized, or you do the real experiment, you do the immunoprecipitation with the biotin, correct? And so immunoprecipitation is very handy in the proteomic field, and they found these genes and the pathway which were related to the Golgi membrane, extracellular exosome, and these enriched in the ataxia disorder. I think that's it, and this is what I do. I don't work on the neurodegeneration, I work on neuroendocrine, and I have done the phosphoproteomics of non-functioning pituitary tumors, and we found significant difference between the recurrent and invasive. Usually people say that if it is invasive it will recur, but there is molecular difference as well, and is like normal to the physiological thing. We found serine as one of the most and tyrosine less, because we did not do the tyrosine PTYR 1000 precipitation, and these are the list of phosphopeptide which are significantly upregulated in the recurrent one. The red thing shows, and Volcano shows that beta catenin, ASHD and ITH2. The drawback of phosphoproteomics is you get mass spec give the information, but then you do not get the antibody. So for validation, either you do the mass spec of different type or you leave it. So I did, out of 3, I did only this one for the validation. I also have done the proteomics of the same data. It's always ideal to do proteomics and phosphoproteomics or whatever PTM you want to do on the same sample. Then you can compare the thing that okay, it is not overexpressed but it is phosphorylated, it is not overexpressed but it is glycolated. So I found a different set of pathways in invasive and recurrent, and there was different upstream regulator for both, which clinical people says that okay, if it is invasive it is recurrent. My answer is no. They slightly disagree with me. And then what? So, and this is the information I get from the Ingenuity pathway, and we say that okay, it's the integrin linked kinase is involved, and I validated with the immunohistochemistry. I'm not going to bore you with this. This I already showed, and this is the PhD student I'm co-supervising. So we also did the phosphoproteome. This is the proteomics in another type of endocrine tumor, the sparsely granulated tumors, and we found the overexpressed protein which are labeled, and then we deduced. So this is whole thing is bioinformatics. The theory is we found TNIP overexpressed and majority of our proteins were underexpressed. TNIP is 111 times overexpressed. So we said with the bioinformatics we deduced the what are the transcription factors involved in the underexpression of protein, and then we find out that TNIP1 is regulating one of the topmost enriched. So TNIP down regulate ELK1, and that may be the reason of down expression of all these 200 proteins. And this is the phosphoproteomics, and thank you. Any questions? Ask anything on proteomics. No? You want to say something? Go on. Any silly question, if anything comes to your mind, maybe I don't know the answer, but you can ask. Anything, shoot for the proteomics. So you know, you can think about experiment, what you learn, what all you can do with the proteomics, the pathways, the mapping, going back and forth from protein to genetic and everything, all you can do. Great, thank you. Okay.